Microbiology Society India. Pharmacology. As you know, in the branch of science, the pharmacology is very important for drug discovery. Apart from this one, it is also pharmacokinetics, pharmacodynamics, clinical pharmacology, toxicology, pharmacogenomics. So, to study the genetic variation, to study the harmful effects of the drugs, and also neuropharmacology, that is the study of the different drugs which affects on the neurophysiology. Apart from this one, this pharmacology workshop will help us to drug discovery and also development in the drug. It ensures the effective medication, use of the different medicines. It also provides the insights of the different disease mechanisms. So it also supports the personalized medicines based on the genetic makeup. So the clinical pharmacology, actually it is the medicine which is useful to apply in different treatments of different diseases. So this workshop definitely benefits to the students and the researchers. So on behalf of the Venkatesh Mahajan Senior College, Department of Microbiology, in collaboration with Microbiologist Society of India, we are conducting this workshop for 2 days. The first day, that is 8th of February, so that is the online mode, and on the second day we are conducting on a offline mode. So actually this workshop is conducted under the Pradhan Mantri Uchchatar Shiksha Abhiyan scheme, that is PM Usha scheme. So welcome to all once again. So good morning everyone. I, Dr. Anil Devkar, on behalf of Venkatesh Mahajan Senior College, Dharashiv, and the Department of Microbiology, we all extend a warm welcome to all our distinguished guests, esteemed faculty members, expert speakers, and enthusiastic participant students. So today we are gathered here for 2-day state-level workshop on the hands-on training of pharmaceutical techniques. This workshop aims to equip the students, UG, PG students, researchers, teachers with valuable pharmaceutical knowledge and hands-on training to enhance their career prospects. So as per Indian tradition, so we are going to initiate our opening ceremony. Before that one, I am to introduce our dignitaries and patrons of our organization. So Honorable Rekha Mahajan, Mentor of Tapasvi Public Charitable Trust, Honorable Poonam Mahajan Rao, Chairperson of Tapasvi Public Charitable Trust, Honorable Advocate Milinji Patil, Secretary of Tapasvi Public Charitable Trust, Honorable Sheshadri Anna Dange, Treasurer, Trustee of Tapasvi Public Charitable Trust, Honorable Dr. Abhay Shahpurkar, Trustee, Tapasvi Public Charitable Trust, Honorable Kamlakar Bapu Patil, Trustee of Tapasvi Public Charitable Trust, and also our Dr. Prashant Chaudhary, Principal, Venkatesh Mahajan Senior College, Dharashiv. So this is our organization team which supports us for organizing this event and also conduct this event smoothly. So now it's time to welcome our guest, chief guest. So, welcome to all.
लाइट्स लावा देखा फैन लाइट्स
Soon they will enter in the auditorium. So welcome them. After that, we will start as per our Indian tradition, the prayer to the Bharat Mata photo, and then we will start our sessions. So welcome to all. So Honorable Advocate Vinji Patil Sahib, Dr. Prashant Vishwi, Director of Dr. Babasaheb Ambedkar Marathwada University, Sub-Campus Dharashi. So welcome to you all. I request all the dignitaries to join us. Have a seat on stage. Venkatesh Mahajan Senior College, Dharasiv. Under the scheme of PM Usha Pradhan Mantri Uchitar Shiksha Abhiyan, Department of Microbiology in collaboration with Microbiologist Society India organized the 2-day workshop. So good morning. So I, Dr. Anil Devkar, on behalf of Venkatesh Mahajan Senior College, Dharasiv and the Department of Microbiology, I welcome all our distinguished guests, esteemed faculty, members, expert speakers and all the students. Today we gathered here for 2-day state level workshop on hands-on training of pharmaceutical techniques. This workshop aims to equip the students with valuable pharmaceutical knowledge and hands-on technical training to enhance their career prospects. As per our Indian tradition, on every auspicious event we begin with a seeking by the blessing of our Almighty. So I now request all the honorable dignitaries to kindly join us and pray for the Bharat Mata photo. I request all the dignitaries. Now we come to one of the most significant moments of our event, that is the felicitation of our esteemed dignitaries. So felicitation is not just a gesture of honor, it is our way of expressing our deep gratitude and appreciation for the dedication and contribution those who guide, inspire and support us. Now I request Dr. Mukran Chaudhary sir to felicitate our Honorable Advocate Vinji Patil Sahib, Secretary of Tapasvi Public Charitable Trust, and today's our President of workshop. Now I request Dr. Prashant Chaudhary sir to felicitate our chief guest, Dr. Prashant Dikshit sir, Director of Dr. Babasaheb Ambedkar Marathwada Sub-Campus, Dharasim. Thank you, sir. Now I request Swaz Patil sir to please felicitate our principal, Dr. Prashant Chaudhary sir. Now I invite our organizing secretary of today's workshop, Dr. S.G. Chaudhary ma'am, to share objectives and vision of this workshop.
Good morning to all. First of all, I express thanks Good morning to all. First of all, I express thanks to our respectable patron members for giving us opportunity to organize such a event. Honorable dais, respectable dignitaries on the dais, president of today's workshop, Honorable Advocate Milind Patil sir, Secretary, Trustee Public Charitable Trust Committee, respectable inaugurator, Dr. Prashant Dikshit sir, Director, Dr. Babasaheb Ambedkar Marathwada University, Sub-Campus, Dharasim, Chief Guest, Professor Dr. A.M. Deshmukh sir, President, Microbiologist Society, India, Dr. R.M. Kukrakade sir, Chief Guest of tomorrow's valedictory function, Professor Dr. P.G. Chaudhary sir, our respectable principal, the respectable guest available online for today's session, Mr. Kakasaheb Kore, founder of IIPTCR Pune, Professor Komal Khatta, pharmaceutical expert, IIPTCR Pune, Dr. Sheetal Chaitanya Pendekar, Associate Director, Bicon Biologics Limited, Bangalore, India, Dipti Jadhav Madam, Team Leader of APR Atlas LLP, Ahmedabad, Gujarat, Professor Shweta Padare Madam, Research Scholar of Krishna Vidyapith, Kannada, and all the participants, faculties from 11 different places of Maharashtra, and all my college staff, teaching, non-teaching staff, and my dear students, all participants. I am Dr. Chaudhary S.C. from Department of Microbiology, Venkateshwara Senior College, Dharasim. I shortly here introducing you with my department and sharing here workshop schedule. My department started in 2009, though it is still a non-grant department. It grants a bright future to our students through various student-centric activities that provide them different opportunities for career development. The purpose of organizing today's workshop on hands-on training on pharmaceutical technique in collaboration with Microbiologist Society India. The purpose aligns with the vision, mission, and objective of our college. This is an attempt to to make our students familiar with different pharmaceutical techniques useful for their future jobs and also to motivate them for research purpose. Here I am declaring the schedule of this 2 days workshop. This workshop is conducted in successive 2 days. Today 8 February and tomorrow 9 February. The first day of the program is conducted completely by online mode. A very few number is present here in function hall physically for inauguration purpose, but I expect near about 150 participants by online mode. So first day started with inauguration, and today successive 4 sessions along with keynote speech are conducted by online mode. First speech is by Mr. Gore Kakasai Jagannath on the topic Pharmaceutical Analytical Techniques and Assay Methods. Then second speech will be conducted by Koval Katar Madam on quality control of finished pharmaceutical products. Then after the lunch session third will be conducted by Dr. Sheetal Chaitanya Pendekar Madam on the topic regulatory requirements for biological product production. And fourth session is conducted by Dipti Jadhav Madam on the topic class number API and pharmaceuticals. All the sessions having valuable topics. I request all the participants, offline, online, those are present, to be present during all sessions. The second day also start with guest felicitation. One guest speech by Shweta Patari Madam on QC of food and nutraceutical analysis. Then all the sessions after the speech are of technical sessions which are conducted by offline mode. It is second day schedule. On second day, one guest speech is there, and all the 4 sessions will be conducted by hands-on training mode by distributing the students in different batches. So it is expected that all the participants must present physically tomorrow here. One more request, all the participants must fill feedback forms today and tomorrow also, which is your guidelines for our program. Also, it is your presenting for the program. So, here is the schedule for 2 days program. I welcome all of you for the workshop and express thanks for being participated in our workshop. Thanks to all.
Thank you, ma'am. Now, we are honored to have Dr. Prashant Dikshit, Director of Dr. Babasaheb Ambedkar Marathwada University, Dharashiv campus, with us today. Now I invite him to share his insights on the importance of this workshop.
Respected dignitaries of the dais, Honorable Advocate Dilip Patil sir, Secretary, Tapasvi Public Charitable Trust, Dharashiv, Principal of this college, Professor Prashant Chaudhary sir, Dr. Suvarna Chaudhary, Head Department of Microbiology, Sumaya Mistry, ma'am, present over here. All the staff members of Venkatesh Mahajan Senior College, the delegates who are present offline as well as online, the chief guest of this function, Professor A.L. Deshmukh sir, President, Microbiology Society, and dear students. At the onset, let me declare that hands-on training of pharmaceutical technique, this workshop is being inaugurated. I also take opportunity to congratulate the organizers because in today's scenario, the pharmaceutical products or pharmaceutical microbiology is one of the most important aspects of human life, and giving a skilled personnel to the society is need of our respected Prime Minister Narendra Modi sir has given us this concept that we should have a learned, skilled manpower so that we can go for independent India in a new fashion, so that we can have our own product made in India. So made in India is the slogan given by our Honorable Prime Minister. So considering this fact, let us come to pharmaceutical microbiology. We as an academician, we are preparing curricula for all these subjects, and especially after introduction of national education policy, we are looking for the skilled courses. And in that case, pharmaceutical microbiology is being considered as one of the most important course as far as employability is concerned. And in that regard, it is mandatory that the students who are learning this particular subject, they should be skilled with various courses related to pharmaceutical microbiology. And this is an important event to be organized by this college, that you are giving an opportunity to all these students to get acquainted with pharmaceutical microbiology, especially the hands-on training is important. Because when we prepare curricula, we put so many things in that curriculum. As far as pharmaceutical microbiology is concerned, we put the basics of pharmaceutical microbiology, what are antimicrobial agents, what are their mode of actions, how those are being produced, either by microorganisms or by chemical processes. And then we have several quality control tests which are to be taught theoretically to these students. But if you look at the actual applications in the pharmaceutical industry, it is found that there is rather a wide gap between what is being taught in the class and what they are to perform in the industry. And this workshop will definitely help students to get an insights of those techniques, what they have to do in industry actually. Now coming to what is being done or what will be done in these 2 days. Ma'am has just now given us a complete schedule for 2 days. I think you are going to have quality control techniques. The experts will be talking on those techniques. If you compare it with the curricula, we just talk about lab tests theoretically. We don't perform those things. Same with sterility. The sterility that is being maintained in the college or university laboratories, that is no way at par with the industrial level. So students need to know how exactly they have to perform in the industry, and that is the motto of this particular workshop. So in that case, it is duty of all the students who are attending offline or online that you should be attentive for these 2 days, get everything known very well by the experts, because so many industrial experts as well as researchers, they are going to provide you insights into all the different aspects of pharmaceutical microbiology. So be attentive, try to grasp as many things as possible, because this is going to help you increase your employability. If you have detailed knowledge of those techniques, and in due course of time, during these 2 days, you will be having hands-on. Of course, those online sessions will also comprise introduction of various equipments that are present in the industrial laboratories. So they will be making So get yourself well acquainted with this current knowledge, because pharmaceutical industry, if you consider it as one of the most important aspects, it is ever growing. It has regulatory constraints, and most importantly, those regulatory constraints are being given by national as well as global levels. Because if you are there in the global market, the pharmaceutical products, if they are to be sold in the global market, we need to follow the guidelines which have been provided by the international agencies. In pharmaceutical industries, you have US FDA approvals, you have some experts from Australia, you have some experts from various countries, even German people, though they used to come over here and they assess your industry. So every now and then, there is always a check. Now why this check is necessary? The reason is very simple. If you consider in past scenario, we had the basic needs of life like food, shelter and clothes. Nowadays, we need to add pharmaceutical products to those basic needs. It is because of the lifestyle that we people are living. If you consider Indian scenario, we have at least 50% diabetic patients. So every day you require one or other pharmaceutical product with you. And if those pharmaceutical products, if they are related with the human health, they need to be prepared in very sensitive manner. And that is the reason why you have different types of audits which have been conducted in those industries. So documentation is second important part which need to be learned at this level only. Because when we perform experiments in the laboratory, we never pay attention to this documentation part. But as far as industrial scenario is concerned, we need to keep an eye on documentation also. So these experts, they will also make you know how the documentation is being made and how it is being maintained at the industrial level. I'll give you the simple example. Many of you are working in the laboratories, college laboratories or university laboratories. You go for weighing, you weigh the material and then immediately you put off the balancing, you go on. There is no mechanism that who has used it, when it was being used, for how much time the instrument was used. In industry, you need to maintain the records for every equipment. So those kinds of things will definitely be provided to you during these 2 days. So finally, I request all the students, because this is really an important opportunity for you people to get a detailed knowledge on pharmaceutical techniques. So be ready for those 2 days, all the sessions, attend them properly, ask questions to the experts, get your doubts cleared, and probably this will help you, rather this will make you employable in coming scenario. Not even the PG students, UG students can also have a chance to join the pharmaceutical industries. So those who are present over here and those who are online, they can think in this way. And through this workshop, I am sure that the students will definitely be employed in future in the pharmaceutical industries. Finally, I thank the organizers for calling me here for the inauguration of this workshop. So thank you very much.
Thank you, sir, for such a insightful, informative talk. Now, the Dr. A.M. Deshmukh, President of Microbiologist Society of India, today, due to some his personal work, he is out of station, so he is not available. But he sent a video message to guide us regarding this workshop. So we will play it on screen. Thank you.
Honorable Principal Chaudhary, other dignitaries present for online and offline program, all attendees attending this program in pharmaceutical techniques, 2 days workshop. I am very happy today to give a keynote address in this 2 days workshop. I know that this workshop is being sponsored by the central government under the scheme of PM Usha. So, Prime Minister Uchchatar Shiksha Abhiyan is a program of central government to promote the science techniques and to build up a science infrastructure. Under this scheme, I first congratulate Venkatesh Mahajan College for conducting this 2 days program on a very, very beautiful topic, pharmaceutical industry. I appreciate also the efforts taken by Chaudhary Madam, Head of Department of Microbiology, and Sumaya Madam, the faculty in microbiology. This workshop is a need of today. The reason is that when India got a freedom in 1947, we were far behind in pharmaceutical industry. We were having the pharma business only 10 crore rupees at the time of freedom. Now, what is the position in India? Now we are 8th in the rank of the world. Meaning is that pharma industry is well developed in India. But still government want to progress, to develop. USA has a 42% share of total pharma industry of the world. Next to it is China, then Belgium, Germany, Switzerland, and many other countries are there. So, in India, there is a tremendous scope for the pharma industry. And whenever we want to develop a pharma industry, we want trained hands. Why trained hands are required? Whenever we are developing a pharma product, to increase the efficiency of the product, to check the purity of the product, to characterize the product, and to increase the production of the particular metabolite, we require trained hands in pharmaceutical techniques. Various pharmaceutical instruments and various pharmaceutical techniques are required in pharma industry. The techniques are based on chromatography. So, simple chromatography, thin layer chromatography, gas chromatography, high then HPLC, HPTLC. Likewise, various types of chromatography are required. Spectroscopy is also required. Double beam spectroscopy, then ultraviolet spectroscopy, then mass spectroscopy, fluorimetry. Likewise, different spectroscopic techniques are required. NMR is also required. Nuclear magnetic resonance spectroscopy. Likewise, many techniques are required in pharmaceutical industry. And Many students do not know the equipments required under Usha scheme. The government is funding to purchase various instruments. Under the government scheme, government is providing money for training of the students. And really, during these 2 days, one day is online and one day is offline, student will learn many techniques. That is called as the hands-on training. Here, I request all the students that be attentive, learn everything. Because whenever I am meeting with the many industries in pharma industry, biotech industry, they are complaining me. They are telling that, sirs, there are many students, but they are not well trained, they are not well learned. We want trained students. Students do not know about the good manufacturing practices. Students do not know about the quality assurance and quality control. So I request all the students, during these 2 days, you take the maximum advantage so that learn many techniques, take experience hands-on training and make yourself fit for job. Jobs are available. You see, once upon a time, it was very difficult to get a tablet, even painkiller, even antacid in India. Now, India is exporting many pharmaceutical products. For example, Reddy's laboratory is there. For example, Serum Institute of India is there. Cipla is there. Ajanta Pharmaceutical is there. Like that, many industries are there which are developing. And these industries want to do many things. They require a trained person. Jobs are available. So I request all the students, during these 2 days, take the maximum advantage and get fit for job. Whenever you will enter into any pharmaceutical industry, your hands are required for the development of pharmaceutical industry, for the development of India. So, as a president of Microbiologist Society, it is my duty to wish all the best for this 2 days program. I know that one of the eminent speaker, Shri Kore from board, he is coming. He is a very well experienced person in pharma industry. Along with the Kore, Shweta Padtare and many other speakers are coming. They will take online and offline program. It is your duty to cooperate them and make this 2 days program successful. Thank you. Thank you very much.
Thank you, sir. Now I take the honor of inviting our Honorable Advocate Minanji Patil, Secretary of the Pacific Public Charitable Trust, to deliver the presidential address.
In participating this unique second program conference workshop what you say, that is 2 days on the subject of hands-on training of pharmaceutical presence of our director, Dr. Prashant Dikshit sir, who is very well known and from this
microbiology department. I am sharing with dice with our principal, who has taken very great initiative and efforts by collecting the entire team of Mahajan College in organizing such a nice function today, of today's function. I also congratulate Dr. Suvarna Chaudhary, who is leading this department since many years, and Sumaya Mistry of
and also alumna of this college. It was really difficult for me to remain present, but by the grace of God and wishes given by our staff members, teaching and non-teaching, by presenting, remain present and in person, have given well wishes for speedy recovery to me. Some of
faculty members meet me here only, but some have personally met me, and due to their best wishes, I remain present here. I am very much thankful for those teaching, non-teaching of college, school, all the faculty members. Really, I was not thinking that this college, Dikshit sir, is capable of organizing such a beautiful function, offline and online, on this subject of microbiology department.
I am very much glad I have asked our principal.
Very much glad I have asked our principal,
Students are present today.
Dr. Chaudhary, that how many microbiology students are present today. He told me that maximum number of students are present. I hope
Who are present today, at least give
that
communication
the
to the
students who are
absent
present today
students.
at least give communication to the absent students to remain at least present tomorrow, or join in hands with the online at least. But what happened, I was discussing with our director Dikshit sir. Nowadays it is a fashion to remain absent in college. That is a fashion. Students, all the students, microbiology and other science, arts, occasional, that they are having spirit to attend the college and respect their parents. I to join microbiology. It was a so nice department and faculty. I like microbiology. Now it is the science I don't know exactly what is the All India कोणतं प्राइज दिलं जातं ते Nobel Prize. Can you tell anybody from this faculty or student that who was awarded the Nobel Prize on microbiology recently, currently? Do you know? I am not having that much information. But to study microbiology, you should have idols of that Nobel Prize winners. And therefore, I remember the students, our ex-alumni, Nazia and Afshan. I think that out of these two ex-alumni, one is doing some research on cancer drug, I think. Is it correct? And therefore, our student ex-alumni of this college is already doing research. I think mostly it is correct in on cancer. It can happen from so. I request offline and online students that there is a great opportunity in this microbiology field. This microbiology field is very important because in this field, what is microbiology? You might have understood. Now, under the new education policy, even physics and chemistry students can shift to microbiology. What I got knowledge about the new education policy. Sir, with great efforts, our Tapasvi Public Charitable Trust has undertaken this non-granted course to our college. But our faculty has proved it that this is a very nice course and they have successfully run this course. I congratulate all the faculty members for that and they wish them that they will do the needful in future years. Trust will cooperate because what is microbiology? I am having 4-5 minutes. I will not give your valuable time. What is microbiology? That is the study of microorganisms which are not capable to be viewed by naked eyes. What I understood. For that, what is required? Dialectic, microscopes. And that study is required. And therefore, under this guidance, this microbiology department, essential in our life on this earth, to play a major role in many aspects of our lives. Particularly, what it includes biochemistry, physiology, cell biology, ecology, evolution, and clinical aspect. I think that today's workshop is on clinical aspect, that is pharmaceutical. And therefore, this microorganism study includes bacteria, viruses, fungi, algae, protozoa. You might have gone through all these words, archaeas and proons. How, lastly, while concluding my contention, how microbiology can be applied in our life? Firstly, in medicine,
can be important. It can create a biofuels. May I correct, sir? Biofuels and clean up on pollution. Microbiology helps to control pollution and create biofuels, and particularly in the area of food, microorganisms can be used to make fermented foods and probiotics. I think that curd, dahi, it is one of the study of the microbiology. How it is important in our life, that is also material. And last but nothing, and major achievement of the microbiology is in the field of agriculture, where sons and daughters of farmers. Our country is lead by the farmers, and microorganisms can be used to grow up crops. Yield can be achieved more with the help of microbiology. Why I am putting all those things before you? Your department is so nice. If you make hard work and participated in such a workshop, you can achieve many things. I request all the students notes. I think that if it is possible, if they are not any writing material, you can provide them. Tomorrow, offline, online, they should draw some notes and participate. Just be told and guided by our director, sir, that you should attend this very seriously. And also our main support, Mr. P.P. Deshmukh sir, Dr. Deshmukh sir, who is President of Microbiology Society of India, has given a major contribution. We have heard on video their keynote address, and he has supported. Now, while attending this program, Dixit sir, Deshmukh sir, Kakasai Kore sir, Komal Prasad sir, Madam Sheetal Chaitanya Peltekar, Priti Jadhav, for today and tomorrow. Also on second day, Dixit sir is also attending, I think. Sweta Bhattare, Kore Kakasai, and Bhubragale sir, they are giving valuable guidance to you. I think that we should have a very great culture of this workshop. Don't participate as a formality. Sir has told us to remain project. Devote this your mind in this workshop, and college has organized this workshop very nicely. I again congratulate our entire team under the guidance of our principal and department faculty, Suvarna Chowdhury and Mr. Madam, and all other staff who are cooperating. Though there is no their subject, but all the teaching faculty of arts and science of other departments have remained present and have shown the interest. Students have shown the interest. Therefore, again, I welcome to all, you all the best wishes for the success of this conference. And I also congratulate and give thanks to the organizing team. Because the advisory committee is there, I will not take the time because the other participants are going to address online.
that they have given me to attend and participate in this inaugural function, giving me an opportunity for that purpose. I give thanks to them. Thank you very much.
Thank you, sir. Such a wonderful, brainstorming, informative talk which is delivered to the all students, participants. Thank you once again. Thank you. Now, as we conclude this inaugural session, it is time to express our heartfelt gratitude to everyone who made this event possible. So, I request Sunya Mistriman, convener of this workshop, to propose the vote of thanks.
Good morning, everyone. I am Sumeya Pharmaceutical from the Department of Pharmacology, present here to express official thanks for the event. I want to express heartiest thanks to our patrons, Honorable Tekamajan Ma'am, Honorable Poonam Mahajan Rao Ma'am, Honorable Advocate Nilin Patil Sir, Inaugurator of the program, Honorable Dr. Prashant Dixit Sir, Chief Guest of the program, Honorable Dr. N. Vishnu Sir, Chief Guest of tomorrow's Validity Function, Honorable Dr. R.N. Khobakar Sir, and respected Principal Sir, Dr. Prashant Chaudhary. I want to express my thanks to respected guests available online for today's session, Kaka Sahib Kodekar, Professor Komal Khatal Ma'am, Dr. Shrital Chaitanya Purnikar Ma'am, Dipti Garo Ma'am, Professor Shweta Kartari Ma'am, all the participants, faculty from different places of Maharashtra. I want to express my thanks to organizing team, teaching and non-teaching staff in making this event possible. Thank you for being here and showing your enthusiasm for learning. This workshop is great opportunity to learn and grow. Thank you.
Thank you, ma'am. Now, there is a technical session. We will start. That will be online mode. The session first will be shown displayed on the screen.
Hello.
Now, this workshop which is being conducted by microbiology department of the Venkatesh Mahajan Forest Mahavidyalaya. This workshop is sponsored by Pradhan Mantri Uchchatar Shiksha Abhiyan, and this workshop is being carried out in collaboration with Microbiologist Society of India. The title of this workshop is the hands-on training of pharmaceutical techniques. For the first session of this workshop, we are having a very eminent research personality or eminent person available with us, Professor Dr. Kakasai Kore. Dr. Kore is presently working in the Raigad Gyanapit College. Now, I am very proud to introduce such a personality who is having around 22 books published. He is having around 19 patents. Already he's got the 19 patents, and he is working on the 9 patents. Then he has published more than 40 research papers. He is a research guide. Then he is a founder member and head of the International Institute of Pharmaceutical and Clinical Research. He is also founder and head of the Job Well India Pune and Core's Technology and Services. He is also working there in another organization, that is Health Sleep India. So we are having a very eminent scholar, research scholar, as well as a very enthusiastic and a very active personality with us, Professor Dr. Kakasai Kore. So I request Professor Dr. Kakasai Kore to start the first session. So therefore, over to Kakasai Kore, sir. Thank you.
Hello. सर आवाज एको होतोय. आवाज एको होतोय. Hello. Hello. Now my voice is clear.
आवाज इको होतो. हा बरोबर
एको होतोय सर आवाज.
येतोय सर.
ओके. स्लाइड दिसते तुम्हाला? स्लाइड शेअर झाली का तुम्हाला? सर स्लाइड शेअर होऊन राहिली. हॅलो. ओके, 1 minute सर.
Yes, sir. Yes. Yes. Your slide is visible.
मॅडम आवाज एको होतोय. तिकडून तुमचा मला जो आवाज येतो ना तो एको होतोय प्लीज.
You start presentation, sir. Your voice is clear. Okay.
1 minute. मला स्लाइड तुम्हाला मोठी दिसेल ना. ओके, जस्ट 1 minute.
सर तुमच्या स्लाइड दिसत होत्या आणि
नाही छोटी दिसत
चालू
होती
करू
स्लाइड
शकता.
ती. स्लाइड छोटी दिसत होते का मोठी दिसत होती तुम्हाला? थांबा एक मिनिट. एक मिनिट हा सर.
हा सर बरोबर. पीपीटी प्रेझेंट करा फक्त. आता तुम्ही शेअर केला
मग
स्लाइड.
ते पीपीटीच एक मिनिट हा.
माझा चालू असला की इको होतो म्हणून ती बंद केली.
हा ठीक आहे, एकच मिनिट सर, जस्ट 1 मिनिट.
Share your slide.
नाही, स्लाइड स्लाइड पूर्ण स्क्रीन शेअर झाली, पण स्लाइड इथं शेअर करायची मला तुम्हाला ना.
स्लाइड ओपन करा सर.
नाही, स्लाइड ओपनच आहे सर, फक्त काय होतंय इथं शेअर करताना थोडस हे. हे मॅक्झिमम होतंय का तुम्हाला सांगा?
सर
हा जस्ट.
स्लाइड शेअर करा सर तुमच्या स्लाइड्स.
हा
हा
स्लाइड,
स्लाइड दिसत आहेत सर तुमच्या.
पण ते
दिसत आहेत. Yes, yes.
मॅक्झिमाइज व्हायला पाहिजे. एक मिनिट सर.
स्लाइड विजिबल आहेत सर.
पण पूर्ण दिसते का तुम्हाला स्लाइड मोठी? छोटी दिसते ना स्लाइड तुम्हाला?
दिसत आहे सर.
जस्ट 1 मिनिट सर. मला ते स्क्रीन पूर्ण स्क्रीन इथं सर पूर्ण करायचं काय प्रोसेस आहे स्क्रीन? नाही सर, मला ते स्क्रीन जस्ट अ मिनिट सर. हॅलो सर. हॅलो.
हा सर.
सर तुम्हाला स्लाइड मॅक्झिमम स्क्रीन मध्ये आता मला स्लाइड मॅक्झिमाइज करायचे, पण ते इथून ही स्क्रीन छोटीच दिसते ना मला.
सर स्लाइड शेअर करा. झाले होते, दिसत होते व्यवस्थित. तुम्ही अनशेअर केलात स्क्रीन परत आणखी.
ते विंडो मध्ये मी ओपन केलं होतं ना, पण माझ्याकडे मला मोठी स्लाइड दिसते ना, ते छोटी दिसायला लागली.
आता फक्त स्लाइड्स ओपन करा सर. शेअर झालेली आहे स्क्रीन. सर
हा जस्ट मिनिट.
स्लाइड शेअर करा की एकदा केल्यास बरं करा.
काय
एकदा स्लाइड शेअर करा सर.
काय होते सर, स्लाइड मी शेअर करतोय तुम्हाला, पण मला इथं समोर ती छोटी विंडो दिसते. मला पण मोठी दिसायला पाहिजे ना विंडो ती. आलं का लक्षात? आता बघा, मी विंडोज मध्ये जाऊन करू का एंटायर स्क्रीन मध्ये जाऊन करू सांगा?
हॅलो
मुलं
सर.
जॉईन आहेत का सर? हा हा ओके.
हो, मुलं जॉईन आहेत सर. 60 मुलं जॉईन आहेत युट्यूब वरती. इथं 60-70 आहेत. तुम्ही हा
एंटायर स्क्रीन मध्ये जाऊ का मी?
हा काय करा, इथं जो मध्ये बॉक्स आहे वरती हेड केलेला ॲरो आहे, त्या ॲरो वरती क्लिक करा.
एंटायर स्क्रीनच्या?
नाही 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 नाही, आता तुमच्या गुगल मीटच्या मधोमध जो ॲरो आहे त्या
नाही
ॲरोला.
नाही, ते मी अपलोड करतोय ना सर. एक मिनिट हा. एंटायर स्क्रीन माझी आताच आलेली आहे. हा थांबा एक मिनिट सर, जस्ट पुन्हा करू आपण ती प्रोसेस. मी तुम्हाला तसं म्हणतो अगोदर. हे बघा, प्रेझेंट नाव म्हणलो. हा
हा
प्रेझेंट
करा
नाव
सर.
मध्ये एंटायर स्क्रीन का?
करा करा सर करा. हो.
हा आता इथे एंटायर स्क्रीन येते, पण हे मॅक्झिमाइज कसं होणे मला इथं?
नाही नाही नाही, करा ना सर. आधी शेअर तरी करा की. माझ्याकडे दिसत नाही शेअर. आधी शेअर करा शेअर.
हा शेअर केली सर. ओके.
थांबा एकच मिनिट सर, येऊ द्या इथं. हा, आता काय करा, आता त्याच ह्याच्यावर ऑप्शनवर परत क्लिक करू नका. आता फक्त पीपीटी ओपन करा. पीपीटी जे आहे ना इथं, पीपीटी दिसायलेत बघा तुमच्या क्रोमच्या बाजूला.
क्रोमच्या बाजूला तुम्हाला
खाली
दिसायला
खाली
लागले.
खाली सर, खाली मेन्यूवरती खाली घ्या. तिथं नका करू क्लिक, तिथं नका, खाली या,
बर. हा,
खाली या. तिथं खाली क्रोम जिथं लिहिले ना, खाली क्रोम आहे बघा.
बाय मिनिट.
क्रोमच्या बाजूला पीपीटी आहे सर, मला दिसते सर. तुम्ही
ही
त्याच्यावर नका
ही
क्लिक
ही
करू,
ही का
तिथं
यू
नका
आर
क्लिक,
प्रेझेंटिंग
तिथं नका
तर
क्लिक करू सर.
इथं इथं कर्सर दिसतोय तुम्हाला सर?
कर्सर दिसतोय सर, तिथं करू नका क्लिक. तिथं पीपीटी खाली आहे सर,
बर.
पीपीटी खाली आहे.
टर्न
खाली घ्या,
कॅप्शन्स.
खाली घ्या, खाली घ्या सर, खाली घ्या. हा,
एक
थांबा
मिनिट.
थांबा
हा
थांबा
हे
एकदम,
थांबा थांबा. हे मला दिसत नाही इथं खाली हो कायच.
थांबा थांबा सर, खाली थोडस खाली घ्या.
नाही, स्क्रीन माझी लास्टच आहे दिसते नाही का? थांबा थांबा, इथं का? ओके ओके इथं, हा ओके.
हा हा हा, पीपीटी वरती क्लिक करा.
झाली तुम्हाला
बरोबर,
स्क्रीन शेअर?
आता स्क्रीन दिसत आहे सर. इथं जे काही ट्रॉफी किंवा ग्लास टाईप एक ऑप्शन दिसतो, त्याला प्ले करा पीपीटीला.
ओके ओके, आलं लक्षात.
हा, आणि दुसरीकडे ऑप्शन
आता
वरती कुठेच क्लिक करू नका सर.
ठीक आहे सर. ओके ओके.
एकच मिनिट
सो
सर, मी चालू करतो युट्यूबवर. काहीच करू नका सर दुसरीकडे.
ओके सर, ओके सर.
हा, चालू करा सर.
Okay, good morning to everyone. So, all of you, I hope this is my session which will be fruitful to every and each students, and that will be fruitful for further career, for further interview. So, friends, theoretical knowledge is a different, and technical aspects are so much different. Today's and tomorrow's sessions need is actually to upgrade you at industry level. Many students have, my voice is clear, sir? Hello? 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 Okay.
सर यस यस.
Okay, okay. हाँ, वही आंसर चाहिए मुझे. So today we are here. One of the important points in case of quality control of microbiology, that is microbiology assays, which will be performed for different antibiotics, different and different vitamins. In addition to that, what type of the skills required for the students when they want to work in a pharmaceutical industry, that must know to you. That is one of the microbiological assay is a test for the finished product. But in addition to that, raw material assay, raw assessment of raw material and assessment of finished group product have an important role in a pharmaceutical industry for microbiology candidates. In case of raw material, starch is there, gum acacia is there, any other raw material which is obtained from the natural source. Water is a major source of pharma industry. In such cases, raw material analysis is necessary. In case of finished products, non-sterile pharmaceuticals like tablet, capsules, ointments, syrups, cosmetic products, and sterile product like all parental preparations like saline solution, injectables like small volume parentals and large volume parentals have a sterility test is mandatory, as well as the endotoxin test is mandatory. Now today we start the microbiological assay of antibiotics. Why microbiological assay is necessary? The microbiological assay is necessary to check the potency of the antibiotic for the standardization of antibiotics, for the standardization of vitamin. Microbiological assay is defined as a qualitative and quantitative determination of chemical compound from a simple or even complex material with the use of microorganism. Friends, generally there are the three types of assays in pharmaceutical industry. One is the microbiological assay, which will be carried out with the help of a microorganism. Another is the chemical assay, which will be chemically determined. And third one is a pharmacological assay, that is determination of the therapeutic effect of any drug. So we go for the microbial assay. It is used to determine the potential of drug in animals or man and monitoring and controlling antimicrobial chemotherapy. So here chemotherapeutic agent means what? Generally, there are the two type of ways for the controlling the disease. One is the process is vaccination, that is called as a prophylaxis. The vaccines are given before the infection and drugs are given after the infection, when infection not controlled by the human body. And for that many antimicrobial agents are used which inhibit the growth of microorganisms, that are called as antibiotics, and in other case some source or growth material for the metabolic activity, enhancing the metabolic activity of human being. Vitamins are required, amino acids are required for that there is assays. Defined in the term used to express the biological activity of sample preparation compared to standard preparation. Then what is the advantage of the microbial assay? It is simple. You can perform in your laboratory. It is specific, means for the specific antibiotic, specific microorganism is used. It here minimum requirement of space, labor, material and time, inexpensive and convenient method. Disadvantage, required microbiologist here because it is a skilled technician. Proper calibration is necessary. Less reproducible changes is greater error sometimes if the technician is not expert. And if any good alternative physical or chemical method is used for performing microbiome, performing or checking the quality and quantity of the drug, then people mostly not prefer this. So many drugs concentration is determined by the chemical assay also. Then microbiological assay of antibiotics. Mostly it will be performed by two methods. One is the cylinder, or it is also called as cup plate method, and another one is the turbidometric method. So in IP is one of the important book which is useful for the all type of analysis of pharmaceuticals, whether it is a tablet, whether it is a capsule, whether it is a syrup, ointment, or any other thing. So all students must be aware for that book that is called as IP, that is Indian Pharmacopoeia. Suppose your product is exported to other countries, then you must be refer. Suppose you export in UK, or you suppose export in America, then that country's book are there. For example, United States Pharmacopoeia, Chinese Pharmacopoeia, Japanese Pharmacopoeia, UK Pharmacopoeia. So these are the pharmacopoeias. It is a very important book for the analysis of the finished products. So all of you know there are the two methods. One is a cylinder plate method, or in other words, in your microbiology lab at the time of graduation and post graduation, you perform that method is called as a cup plate method. Another method is called as a turbidometric method. So these are the following medias actually required for performing the microbial assay. So one of the important point here, most of the UG or PG students, the students suppose reach to any pharma industry for interview at that time, most of the students answer is in this case, in case of the which media used for the microbial assay, most of the students answer is nutrient agar or mechanist agar. Friends, this is the composition, and here about 10 medias are here. It will be in IP, it will be marked as A, B, C to up to J, and which possesses the peptone, yeast extract, whip extract, pancreatic digest, dextrose, papic digest, agar, glycerine, polysorbate 80, polysorbate 80, sodium chloride, disodium hydrogen phosphate, and potassium dihydrogen phosphate, and final pH it will be given in this chart. So it is very important. Suppose that company produces any antibiotic at that time, this chart is very important for the question which media, what is the composition of media for performing microbial assay. Then buffer solution is also important. So different pH buffer solution it will be prepared by the dipotassium hydrogen phosphate, that is K2HPO4, and KH2PO4, that is potassium dihydrogen phosphate, and it is a basic of your degree as well as the post graduation at the time of post graduation. So it is very important. It is very important which type of the buffer used in microbial assay. Then another point is the preparation of standard. Standard drug is required when you performing any type of assay, it will be compared with the standard. So for example, 2 milligram, 4 milligram, 6 milligram, 8 milligram, 10 milligram, or 10 units, 20 units, 40 units, 60 units. In this one, this is one of the example. So standard must be prepared in a standard manner, and actually your unknown must be in between that. So when you performing microbial assay, at that time, these two components must be in your hand. That is a standard drug as well as for which you want to check the or which you want to do the performing microbial assay. So this is the stock solution and test solutions used for the preparation of standard preparations. So for example, amikacin is there, amphotericin B is there, then bacitracin, neomycin. हा चार्ट दिसतोय का सगळ्यांना? इज इट क्लियर? Hello everyone. Okay. Amikacin, amphotericin B, these are the number of antibiotics are here. Which method you perform? Is for example, amikacin you must be perform the turbidometric method, that is method B. While for amphotericin you must be perform, it will be performed by the method A, that is couplet method. And here, which solution or which solvent is used to dissolve that drug, it will be given here. That is water, dimethyl sulfonate, 0.01 molar HCl. Then B6 means buffer number 6, B1 means buffer number 1. So these are the different type of the solvents used to dissolve this. And final concentration, you must make that is 1 milligram per ml, 100 units per ml. So concentration of drug, it will be actually in two type of the milligram or in units. Then final diluent for the test solution also given here. This is the standard. So final diluent will be given, and median dose means actually average, it will be given here. Then next point is the preparation of a test sample. Formation and information available for the substance under examination that is unknown, assigned to it an assumed potency per unit weight or value. On this assumption, prepare means you can prepare the whatever the standard as well as test sample in a standard manner. One another important point, in case of the microbial assay of antibiotics, suppose mostly that question is asked to student, which microorganism is used for microbial assay of cannabis sulfate or carbanicillin, amikacin, tetracycline. So most of the students, our answer is in this manner: Staphylococcus aureus or Escherichia coli, or 2-3 microorganisms. For that, that student is familiar. But actually, for in IP, that is in Indian pharmacopoeia, or in US pharmacopoeia, or each country's pharmacopoeia, there is a standard the chart given, and as per that standard, that microorganisms only used for specific microorganism used for specific drug. Here you observe carbanicillin for Pseudomonas aeruginosa. In addition to that, the ATCC number, ATCC means, I think everyone know, that is American Time Culture Collection. That strain only used for example, bleomycin is there, and for that Mycobacterium semigametis, whose ATCC number is 607, which is used there. It is very important for microbiology student. For which antibiotic, which is a perfect microorganism for carrying microbial assay of antibiotics. Same for the vitamin, have a different microorganism. So you don't answer only Staphylococcus aureus or Escherichia coli. So list must be with you, because this list and media, suppose question on the microbial assay at that time, basic requirement is there, what is standard media and standard microorganism for performing the microbial assay. So here nystatin is there, for Saccharomyces cerevisiae microorganism, for oxytetracycline Bacillus cereus is there, for polymyxin B, Bordetella bronchosepta, then tetracycline, Bacillus cereus. The number of examples are here. So you must be refer this chart, and this chart will be given in pharmaceutical microbiology book, or it will be majorly this chart referred from the Indian pharmacopoeia. Then next one is the preparation of inoculum. Inoculum is which is a test microorganism used for that microbial assay. That microorganism grown in laboratory, and sufficient inoculum is developed. So specific media is used there. One of the important property of the microorganism used for microbial assay of antibiotic is that that microorganism must be sensitive to that antibiotic. Vice versa, in case of vitamin, the criteria is that that microorganism must be exotroph for that vitamins. Without vitamin, that microorganism unable to grow in common media. So here, point of the inoculum preparation. Inoculums is the mixture of microbes along with the culture media in which it is growing. But here, one of the important point is the good quality media is required. Standard microorganism like Staphylococcus aureus, Bacillus subtilis, Mycobacterium semigametis, or whatever the microorganism which is enlisted in pharmacopoeia, or next before that whatever the slide is there, as per that slide, that microorganism must be available with you. Then incubate that slant on suitable media. Then using a 3 ml of slant solution, wash the microbes from agar slant. Further, it will be down to larger surface medium such as rocks bottle is used containing 250 ml of agar media. Incubate for 1 day at the required temperature. That temperature is called as optimum temperature, which is better for the growth of microorganism. Wash the growth from the nutrient surface using 50 ml of saline solution, and store the test microbes under refrigerator. Then next one is the methods actually. A cylinder plate or cup plate method. There are the two popular methods are there: cylinder plate or cup plate method, and another one is a turbidometric or tube assay method. In turbidometric method, we are using a liquid media While in cylinder plate method or cup plate method, we are using a solid medium. Most popular at college level is a cylinder plate or cup plate method. A previously liquefied media with the required quantity of microbial suspension is inoculated. The suspense is added in the medium at a temperature 40 to 50 degree and inoculated medium is immediately poured. Or in other words, in a college, we are doing the two methods. First, you prepare a base agar of a suitable media, that is 10 media is given in IP actually. And above that, we pour a seed agar, or that is called as soft agar, in which you inoculate the equal quantity of each tube of same microorganism and pour on the surface of media, that is a soft agar, whose agar percentage is near about 0.8 to 1%. And in that, you mix the microorganism and pour on the surface of sterile base agar in a petri dish. Wait up to cool, and then with the help of a borer, which is made up of stainless steel, sterilize that borer and make a well in a petri dish. So this is actually a routine method. A solution are applied surface of the depending upon some people inoculate the media inoculate the microorganism in the main media. Some people use the soft agar. So here you observe some images. Here Bacillus cereus microorganism is used there, and here only checking standard is there, DMSO is there. So, but this is not a part of this, is a different. Once zone will be obtained, means before that, I will go for okay. First slide, just 1 minute. Okay, here you observe central plate is there, you observe only. In this case, what? Different zone of inhibitions are observed. The disk method is there. Tomorrow we perform the actually well method. So 5 standards it will be given in a different way. You add that different concentration of antibiotics in a different way. But listen, one of the point here, in each way, volume of antibiotic is the same, but concentration is a different. Volume of antibiotic is the same, but concentration is a different. Which, for example, 20 milligram per ml, you add 50 microliter. Do not overload the drug because it will be sprayed over the plate and your experiment will be failed here. So because of this reason, sufficient quantity or exact required quantity only put into the well. So first, for example, 10 milligram per ml, 20 milligram per ml, 40 milligram, and before that you label each well from the back side of the petri dish. There are the 5 concentration, and whatever the last one is there, in that you put unknown of the same concentration. One of the important unknown of the same value, not a concentration, same value, because you want to analyze the concentration of that unknown. Then put that petri dish into refrigerator for the proper diffusion of the drug, and then remove the petri dish from the refrigerator and put into an incubator as it is. Do not invert it. Generally, we, for the growth of microorganism, inverted plate method is there. But here you are performing the microbial assay, and because of this reason, you kept the plate in incubator at an optimum temperature. Depending upon that type of microorganism, you can maintain the temperature which is better for the growth of microorganism. So here one of the very important point, equal volume in each way, but concentration is a different. Suppose 5 different standard concentrations are there and one unknown is there, so you can triplicate or duplicate, you can perform this experiment. Well, another point, the concentration of microorganism first you prepared, and that concentration only used in all plates. Suppose plate number 1, 2, 3. Suppose same microorganism required, same concentration is required when you perform. Not a different microorganism, not a different concentration of microorganisms. And after that, after adding the antibiotics in the well, then incubate the put the kept the petri dish into the fridge for 15 minutes for better diffusion of the drug, and then plate incubate into incubator for 24 hours. Once you remove the petri dish, tomorrow you observe zone of inhibition depending on the concentration of drug in the well. Then another question many times asked to you, how will you determine how will you determine the diameter of zone of inhibition? So in this figure also, whatever the scale is shown here, but actually suppose you have question in this manner, your proper answer must be vernier caliper or antibiotic zone reader. Basically, vernier caliper instead of scale, vernier caliper is correct method for determination of diameter of the zone of inhibition. So here what zone of inhibition it will be observed. So for example, in a 20 milligram, smaller zone is there because drug will be diffused, but finally the drug will be further. Why not inhibit the growth of microorganism? Because at that level, the drug finally diluted and further it has not a capacity to inhibit the growth of microorganism. Because in agar media, there is near about 98% water. So drug will be diffused. Another drug, suppose 80 milligram per ml concentration, it will be diffused more where concentration up to inhibitory level, it destroys the microorganism, and further when it will be diluted, the microorganism is not destroyed. So its inhibitory concentration also very important in initial level. Then you must be plot the graph. So here concentration of antibiotic like 20 milligram per ml, 40 milligram per ml, 60 milligram per ml, 80 milligram per ml, and 100 milligram per ml. And on another axis, that is a diameter of the zone of inhibition, means whatever the zone you observe. So for example, 20 milligram, I suppose diameter is a 2, instead of 2, suppose for example 8 mm, then next will be 1.4 mm. Suppose 2, 4, 6, 8, 10 reading is there. And unknown is 7. So you plot in this manner. उसके बाद यहाँ आएगा. So that is the concentration is 70 milligram per ml. Unknown concentration is a 70 milligram per ml. One of the important point here, your unknown concentration generally must be in between standard range. That is median or not median, but from 10 milligram से 100 milligram के बीच में आना चाहिए. So that will be better for the plotting the graph. If concentration, unknown concentration is more than 100 or less than 10, then determination is again complicated. And because of this reason, you can design or you can prepare a standard approximately more and less than your unknown. So here cup plate method is over. Here cup plate method is over. So you have any question? Then I will go for the another method. Okay. Hello, my voice is clear?
Yes, sir, it is clear.
Okay. First of all, I want to discuss, or can I continue this and then we discussion session?
Sir, okay, sir, continue, sir.
Okay. So another method is the turbidometric or tube assay method. One of the important point is there, turbidometric method have a shorter incubation time.
You can go ahead, sir.
Okay, okay. In case of turbidometric method, it is a shorter incubation period as compared to the cup plate method because for better observation of zone of inhibition, we require minimum 24 hours. Means today you perform and tomorrow generally we observe. In case of turbidometric method, because it is a broth is there, one of the important point is there, microorganism as like human being drinks any, for example, any soft drink is there easily as compared to the solid food. Here microorganisms accumulate food faster, microorganisms accumulate liquid food or broth material more easier than the solid material. One another thing is there, in a broth we can observe the growth after 3 to 4 hours because generation time is a shorter in case of microorganisms, suppose 10 minutes, 12 minutes, 15 minutes, depending on the type of microorganism and depending upon the media condition you maintain there. So within 3 to 4 hours, you observe some turbidity in case of the test tubes, and with the help of turbidometer, we can determine. Then one another thing that is advantage shorter incubation period for the growth of test microorganism, that is a microorganism used for the assay. Disadvantage is the presence of solvent residues, inhibitory substances affect more. Not recommended for cloudy and turbid preparation. Means if you prepare the media which is used for that assay, it is a cloudy and turbid. In such case, this method is unsuitable because whatever the media want to tell, any media is a cloudy already or turbid that interfere on your assay result, and because of this reason, it is not used there. Well, another point is there. This method is rapid, so series of test tubes you must be prepared. Method of determination of standard is the same standard methods you can standard antibiotics you can use. That is one another point is the standard, and suppose you perform penicillin assay, then penicillin is used as standard. Suppose you perform tetracycline assay, in both cases, then tetracycline is used as a standard. Not in this manner, you perform assay for the tetracycline and standard you used suppose gentamicin. So same standard and same test sample must be used. Then 5 different concentration of standard solutions are prepared by preparing the standard curve. Then that standard curve means what? You can prepare a different concentration, that is a 10, 20 milligram, 40 milligram, 60 milligram, 100 milligram per ml, and you can measure the absorbance or transmission first. So that is a standard curve line. Nahi to better way you go for the ultra UV spectrophotometer, where you can determine the lambda max. Then 1 ml of each concentration of standard solution of the sample solutions are placed in each of the tubes in duplicate at 9 ml of nutrients medium previously seeded with the appropriate test microorganism. So different concentration in the test tubes, unknown having his own concentration, whatever I don't know or we don't know, and same amount of microorganisms are inoculated in the same media used for performing that assay. Here you observe, suppose for example in 9 ml blanks is prepared, 9 ml of bacteria with the nutrient medium to all the test tubes. So here you observe where less concentration of drug, that test tubes after incubations more turbid, then further less turbid, then further less turbid, and finally there is no growth of microorganism. So we determine the turbidity of the solution, concentration increases, then turbidity decreases. So concentration increases, turbidity is inversely proportional of that. जैसे जैसे concentration बढ़ जाता है तो turbidity कम हो जाती है. वाइस वर्सा विटामिन में क्या होता है, जैसे जैसे concentration बढ़ जाता है growth is more, and less concentration less growth is there. So this is a turbidometric assay. Graph also prepared as per this turbidity observation. 5 tubes containing the inoculated culture medium with standard drug with the specific dose and test organism. While 5 tubes containing culture medium with test organism and the test sample with different dosage. So here 5 tubes containing inoculated culture, the region behind this most pathogenic microorganism, human pathogens, grow at a 37 degrees centigrade for 3 to 4 hours. Then growth of the test microorganism is measured and determining the absorbance at 530 nanometer, that is wavelength of its against the blank. जो पहला वाला ट्यूब है ब्लैंक चाहिए, उसमें मीडिया है, then there is no microorganism. The standard calibration curve is prepared and the absorbance obtained from sample is plotted on it to obtain the concentration of the test antibiotic. So this is actually methods. One is the couplet method, another is a, but mostly students know couplet method and turbidometric method, but they perfect don't know which media used for the antibiotic assay, which microorganism used for the antibiotic assay, how standard solutions are prepared, how standard microorganisms dilution means culture preparation is there. So that point is very important. Well, another point is there, culture standards are, the numbers are not only for ATCC also used, in some cases another standard labs culture also used there. For example, ATCC means American type culture collection. One another is NCTC. One another is NCIB. National NCIB means National Collection of Industrial Bacteria. NCTC, National Collection of Test Culture. So these are NCTC, NCIB or ATCC. In India also NCL maintains standard culture which is in Pune. So anyone wants the standard culture, you got from that NCL. Okay, so these are the spectrometer for absorbance. In first case, in case of curve plate method, you determine zone of inhibition diameter, and in another way here you use turbidometric or you use the absorbers. Concentration increases, absorption increases. Here you observe this is a source. So after source there is one of the filter is there, and which pass a monochromatic light, that is which have a single only one wavelength. Whatever the normal tube light, normal bulb is a polychromatic light. So further the cuvette is there, or cuvette having one of the bracket is there. When you put a black body, then 100% transmission will be blocked. When you remove the black body, 100% transmission. And when you put the test tubes, blank test tube, blank solution in that, at that time you determine, you can, it's a baseline light, so you maintain 100% transmission when blank solution is prepared. And when further different concentration, same cuvette is used, but one by one, different concentration, different test tube solution you can measure. Here, higher the concentration, less the concentration of drug, more the growth is there and more absorbance of light. Then in second, slightly concentration increases, less absorbance. In third, again less absorbance. In next two, there is again less absorbance, more transmission is there. So you can determine the percent transmission or percent absorbance. And before that, you with the help of blank, you get 100% transmission. Okay. And this is a graph. Means different concentrations are here, and this is a graph will be plotted, and you determine this is a standard graph, and you can sample also plot on that. So this is about the microbial assay. So now you have any question? Hello, sir. Hello.
The students are joined on the YouTube.
Okay. Okay.
So therefore, we will not be able to get the
Yes, sir.
questions.
Okay, sir. Okay, no problem.
The students are joined on the YouTube. So therefore, we will not be able to get the questions.
Okay, sir. Great. Just I want to stop this slide sharing. Can I stop this slide sharing? Again, I will show on another slide of the same also, which is similar. Hello?
Sir, your screen is visible here. You can share.
No, I want 1 minute, sir. But I want to stop this presentation and I will share another. सर दूसरा शेयर हो गया स्लाइड
Yes, sir. Yes, sir. It is visible.
बट इट इज अनदर ना नॉट सेम ना
No. It is second one, not previous one.
Okay. So same slides are there. In this case, generally we determine the inhibitory effect of the drug on specific microorganism. That is a qualitative. Quantitative means in what is the concentration required to inhibit the growth of microorganism. That is the qualitative and quantitative difference in case of the assays. So in case of the qualitative MIC is determined minimum inhibitory concentration. Quantitative we determine the concentration of drug in a standard preparations or in a final formulation, that is finished product. Why it is a need of the antibiotic assay? It is necessary to assay antimicrobiological agents for determination of its potency, for determining the pharmacokinetics of drug in animals or man, and for monitoring the controlling antimicrobial chemotherapy. Why concentration? So one of the thing is there, we determine the concentration, so we check whether that concentration as per label it's okay or not. But before that, when any new drug which will be developed, so it undergoes the preclinical study, means animal study. That clinical study means on human study. At that time, whatever the concentration of drug which is used, that drug does not create any toxic effect on the human being. So concentration must be in this manner in the body of human, which is inhibitory to the microorganism, but at the same time, it does not create any adverse effect on the human being. So any drug should have effect or control that microorganism, but at the same time, it must be safe. So it have a chemotherapeutic importance, but at the same time, drug must be safe. The method is a simple, specific, inexpensive, and convenient, very useful for determining changes in the potency of antibiotics and their preparation. Microbiologic assay of antibiotics, we can determine the zone of inhibition in case of microbial assay. The inhibition of microbial assay growth under the standardized condition may be used for demonstrating therapeutic efficacy of antibiotic. The microbiological assay is based upon a comparison of the inhibition of growth of microorganisms by measured concentration of antibiotics to be examined with that produced by known concentration produced by known produced by known concentration of standard preparation of the antibiotic having a known activity. The test solutions requirement, standard solution and inoculum. Three important points are there. हमारे पास टेस्ट सॉल्यूशन चाहिए जिसका हम अननोन का कॉन्सेंट्रेशन निकालना चाहते हैं. Then another one is standard solution and third one is preparation of inoculum. Next one, media used for that assays already explained with you. Media required for the preparation of microorganism inoculum are made from different nutrients. So, for example, for the growth of microorganism initially or for the growth of test culture, media must be better. Here also media must be good quality. If poor quality media used there, so that what happened? When you poor quality media is used there, that poor quality media itself make the big culture. At that time, zone of inhibition is more, or culture is not grown properly. And because of this reason, standard media is required for the growth of microorganism. So these are the same media. The preparation of standard. A standard preparation of in an authentic sample of the appropriate antibiotic for which the potency has been. A standard preparation is an authentic sample. What is authentic sample? Authentic sample means same antibiotic standard sample is necessary, and which will be obtained from the it will be obtained from the standard source. If standard is contaminated or if it is adulterated, then your result will be wrong. And because of this reason, authenticate sample of the standard, and that is also checked in industry by different methods. Then a standard preparation is an authentic sample of the appropriate antibiotic for which the potency has been precisely determined by reference to the appropriate international standard. The potency of the standard preparation may be expressed in international units or in microgram per gram per milligram of the pure antibiotic. The dissolved quantity of the standard preparation of a given antibiotics in the solvent means different solvent is used for the different antibiotics. Dilute the preparation to get the required concentration as stated and stored in the refrigerator. Usually prepared in the ratio of 1:1 as to 5. This is the solvents used for the preparation of standards. Then preparation of test sample. Similar method is used for the preparation of the test sample also. Then these are the microorganisms which is required here. ATCC only given, but ATCC, NCTC and NCIV source, it is a source of test organism also used. Then preparation of inoculum, that is over. Cylinder and turbidimetric cup method. So its principle and procedure is given to you. This is different concentration and the zone of inhibition it will be. Here, one thing is wrong, not ml in this slide, 0.6 milligram per ml, 0.4 milligram per ml, 0.2 milligram per ml, 0.8 and 1 milligram per ml. Here you observe. So, here 0.6 milligram per ml zone is a smaller one, 0.6 is again larger one, 0.2 is the smallest, 0.2 milligram is the smallest zone, black is there, it is the zone of inhibition. And centric, that is a disc or bell is there. Okay. So here zone of inhibition is less. Next one is again increased, and finally it is bigger. And unknown. Unknown is more than 0.2 ml or it is also more than 0.4, but in between 0.6 to 0.4 or some time it will be here as like 0.2. जितना दिखता है उसके हिसाब से हो सकता है. So ये तो आपको क्लियर होना चाहिए. This is a plot. This is a concentration versus inhibition. Concentration of antibiotic versus zone of inhibition. Zone of inhibition is determined in mm. Then next one is a turbidometry. So same slide is here. So go for the microbial assay of vitamins. So vitamins is a different, which promote the growth of microorganism. Vitamins are important, which are the growth factor needed for growth and multiplication of microorganism. They are very sensitive to small amounts for the growth as a growth factor. Its ability of the test microorganism to synthesize the factor being assayed that form the basis microbiological assay of vitamins and amino acids are performed. Here titrometric method and turbidometric methods are there. One of the important point is there microorganism which is used for the microbiological assay of vitamins. That microorganism must be exotroph. All of you know autotroph and exotroph. Autotroph itself synthesize all vitamins. Exotroph are deficient for the synthesis of a specific amino acid or specific vitamins. So here clean 10 test tubes. So which will be and point 0.5 0 to 0.0 to 0.5 ml, 1 ml, 1.5 ml, and 5 ml respective standard. The cyanocobal, that is cyanocobalamin solution is added. To each test tube, 5 ml basal media stock solution is added. Adjust the final value of the test tube 10 ml. Then other 4 test tubes add 1 ml, 2 ml, 3 ml, 4 ml respective test solution to be assayed. To each test tube, add 5 ml basal medium stock solution, adjust the final value 10 ml by using water. So after that, it will be everything will be sterilized at 121 degrees centigrade for 5 minutes. Here I observe 5 because not more than that, because there is the vitamins, mostly vitamins are destroyed at a higher temperature. After sterilization, cool all test tubes up to room temperature and inoculate with 1 drop of inoculum, that is bacterial culture, which is a test culture. Incubate the test tubes for 64 to 72 hours at 30 to 37 degrees centigrade. Titrate the contents further of each tube with 0.05 normal NaOH using 0.1% weight by volume chromothermal blue as an indicator, which can convert it into green color and determine the average of titration values for each level of standards. Plot the graph and you determine the type curve, same as like. So this is, and another one is the turbidometric method. So here in case of turbidometric method, concentration of vitamin. In first case, suppose concentration of vitamin is 10, then suppose 20 milligram, 40 milligram, and then 60 in this manner. Concentration increases, growth increases. That is more turbid, and because of this reason, you can determine the means less transmission is there. So in this manner, you can determine the turbidity and plot the graph. So here one of the determination of MIC of liquid dilution. ये MIC भी मैं आपको बता देता हूँ. MIC क्या है? मिनिमम इनहिबिटरी कॉन्सेंट्रेशन. जो ड्रग है उसकी स्ट्रेन डिटरमाइन करने के लिए इनहिबिटरी डोज डिटरमाइन करने यूजफुल है. Okay, this is about the microbial assays mostly, and in short I will take these vitamins. So this is my session. Now session is open for discussion, but if it is possible, okay.
Thank you, sir. On on behalf of Venkatesh Mahajan, Senior College Dharashiv and Microbiology Society India, I would like to sincere thank to Dr. Kakasai Pore to give this wonderful sessions on pharmaceutical analytical techniques and bioassay. So I'm again very thankful to you, sir. So this session ends here. So I hand over the next session to Dr. Chikle, sir.
Hello.
Thank you, sir. Thank you.
Thank you, sir. Thank you, sir. My request to all students, tomorrow everyone will be joined there. Okay. And today also all sessions.
Yes, sir. Yes, sir. Yes.
Okay. Okay. Thank you, sir.
Thank you, sir. Thank you.
हॅलो. खडके सर आवाज येतोय? खडके सर. हॅलो.
हा सर, आवाज येतोय सर, येतोय सर, येतोय.
ओके सर, ओके ओके
आवाज येतोय सर, येतोय.
ओके. So good afternoon and welcome
येतोय सर,
all.
बोला तुम्ही.
Okay. Good afternoon and welcome all in this session. Tapasvi Public Charitable Trust Yevtis Venkatesh Mahajan Senior College Dharashiv and Department of Microbiology in collaboration with Microbiology Society India under PM Usha scheme organized in this 2 days state level workshop on hands-on training of pharmaceutical techniques. I am Dr. Jaykumar Shamraj. Welcomes all the participants, students, and professors in this session. In this second session, our guest is Komal Dada Khatal. Her qualification is M.Pharm, specialization in pharmaceuticals from pharmaceutical expert. That is, ma'am is pharmaceutical expert, IIPTCR, Bhor, Pune. So welcome, ma'am, in this session. Once again, I welcome all the participants and I request, ma'am, please start your presentation.
Thank you, sir. First of all, hello everyone. Myself Komal Khatal, and today's session, sir, slide share kona first of all. Slide is visible or not, sir? Hello, sir. My slide is visible or not?
नाही, अजून तर दिसत नाही. अभय सर, हॅलो. स्लाइड नाहीत ना शेअर झाले अजून?
हॅलो.
हा, कोमल मॅडमची स्लाइड शेअर झाली का?
नाही सर, नाही अजून झालेली.
हा.
ओके, आता झाले.
First of all, hello everyone. Today's session is on quality control test for tablet. That is pharma finished product tablet. So first of all, we should know the what is tablet, what are the advantages of tablet and its disadvantages. Tablet म्हणजे काय आहे? Tablet is a compressed solid dosage form containing medicaments with or without excipient. टॅबलेट ही एक कंप्रेस केलेला सॉलिड डोसेज फॉर्म आहे. त्यामध्ये आपण ऍक्टिव्ह फार्मास्युटिकल इन्ग्रेडियंट म्हणजे एपीआय ऍड केलेला असतो विथ और विदाउट एक्सिपियंट. सो अकॉर्डिंग टू इंडियन फार्माकोपिया,
कोमल मॅडम, हॅलो हॅलो. कोमल मॅडम,
येस,
तुमचा स्क्रीन जो आहे ना, किंवा तुमचा मोबाईल याच्यावर ठेवा, ऑटो रोटेटवर ठेवा, म्हणजे तो स्क्रीन थोडासा मॅक्झिमाइज होईल. आणि प्रेझेंट करा. एक आयकॉन बटन दिसतंय बघा प्रेझेंटेशनचं, तिथं प्रेझेंट करा त्यातून. हा, ऑटो रोटेट मध्ये ठेवा. नाही, हा इथून बरोबर राईट.
कोमल लॅपटॉपवर नाही घ्यायचं होतं ना.
हॅलो मॅडम, आणि स्क्रीन रोटेट करा मॅडम, लँडस्केप करा त्याला, पोर्ट्रेट लँडस्केप थोडस रोटेट करा स्क्रीन.
ठीक आहे सर, आता दिसते का?
ओके मॅडम, येस येस. आणि सर मॅडम
प्ले करा
इथे एक
मॅडम स्क्रीन,
छोटा
म्हणजे
टीव्हीचा
पीपीटी प्ले
आयकॉन
करा.
दिसते, त्याला क्लिक करा.
कोमल मॅडम,
हा
तुम्हाला मोबाईलवर पॉसिबल आहे का हे सगळं? नाहीतर मग मला पाठवा ते, मी इथून फक्त मी काय करतो, स्क्रीन हा दिसते, आता दिसते, आता व्यवस्थित दिसते, चालेल. येस 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 सर, आता दिसते ना व्यवस्थित?
नाही मॅडम, बरोबर आहे. आता चालू करा मॅडम प्रेझेंटेशन.
ठीक आहे
ठीक
मॅडम,
झालंय,
प्लीज
आता काही
कंटिन्यू,
प्रॉब्लेम नाही, चालू करा.
प्लीज कंटिन्यू,
व्यवस्थित
प्लीज
दिसत आहे.
कंटिन्यू. हा ओके, नो इश्यू.
सर, आता दिसते ना व्यवस्थित?
येस येस
दिसत आहे, दिसत आहे व्यवस्थित.
येस.
What is tablet? According to Indian Pharmacopoeia, tablet solid, fat, or bicarbonate. That is unit dosage form prepared by compressing a drug or a mixture of drugs with or without diluents. That is without excipients. Okay. And what are the advantages of tablet? Tablets are easy to administer. आपल्याला ऍडमिनिस्टर करण्यासाठी इझी असतात. देन इझी टू डिस्पेन्स, आपल्याला त्या एखाद्या पेशंटला द्यायला किंवा डिस्पेन्स करायला त्या इझी असतात. देन मोर स्टेबल, टॅबलेट्स आर मोर स्टेबल दॅन अदर डोसेज फॉर्म, दॅट इज सिरप, ऑइंटमेंट्स. This other than dosage form, tablet is more convenient. So tablet is more stable. Accuracy in dose, bitter and nauseous substances can be easily dispensed. जे काही बिटर डोसेस असतील किंवा जे नॉसियस सबस्टन्सेस असतील ते टॅबलेट थ्रू आपण इझिली डिस्पेन्स करू शकतो. अँड इम्पॉर्टंट इज टॅबलेट्स आर इकॉनॉमिकल. ओके. नेक्स्ट इज डिसएडव्हान्टेजेस ऑफ टॅबलेट. टॅबलेट्सचे डिसएडव्हान्टेजेस काय आहेत? प्रॉब्लेम विथ कंपॅरिझन टू और प्रॉब्लेम विथ कंप्रेशन टू क्रिस्टलाइन
नेक्स्ट
ड्रग.
स्लाइड.
जे काही क्रिस्टलाइन ड्रग असतील त्या ड्रग्सला कंप्रेस करायला प्रॉब्लेम येतो. देन नेक्स्ट इज हायग्रोस्कोपिक ड्रग्स आर नॉट सुटेबल फॉर कंप्रेस टॅबलेट्स. हायग्रोस्कोपिक ड्रग्स म्हणजे काय? ज्या ड्रग्समध्ये वॉटर ऍब्झॉर्ब करण्याची ॲबिलिटी असते, असे ड्रग्स आपण कंप्रेस केले तर ते सुटेबल नसतात. देन नेक्स्ट इज ड्रग्स विथ लो और पुअर सोलिबिलिटी, स्लो डिझोल्युशन मे बी डिफिकल्ट टू फॉर्म्युलेट. Next is cost of production may be increased because of coating and encapsulation to remove bitter and unpleasant taste. The next is swallowing is difficult, especially in children and unconscious patients. Okay. The next is important is quality control test of tablet. So which are quality control tests are performed? Thickness and diameter, weight variation, hardness.
मॅडम प्लीज, मी थोडस इंटरप्ट करतो. कॅमेरा स्टार्ट केला तर व्यवस्थित दिसेल. कॅमेरा स्टार्ट करा मॅडम.
Okay.
मॅडम कॅमेरा म्हणजे तिथून नाही, तुमच्या मोबाईलचा नाही करायचा. इथं गुगल मीट मधूनच आहे मॅडम. इथं गुगल मीटचा कॅमेरा स्टार्ट करायचा.
ऍक्च्युली मी गुगल मीटचाच करते.
कोमल मॅडम, हॅलो कोमल मॅडम, तुम्ही मला ते मेलला पाठवा, मी इथं डाउनलोड करून इथं एक एक स्लाइड करतो.
ठीक आहे, चालेल.
हा. मीनाक्षी मॅडम,
मॅडम अडचण आहे का मॅडम? काही नसेल तर राहू द्या मॅडम, कॅमेरा नका स्टार्ट करू, स्क्रीन शेअर करा फक्त, राहू द्या.
ओके सर.
आवाज मॅडम, आवाज येत नाहीये तुमचा.
मॅडम मॅडम कंटिन्यू करा मॅडम.
आवाज मॅडम, आवाज येत नाहीये.
माईक ऑफ आहे का तुमचा? माईक ऑन करा आणि कंटिन्यू करा, स्लाइड दिसते.
Okay, quality control test of tablet. In that, first one is thickness and diameter. Then next is weight variation. Then hardness, friability, drug content, disintegration time, and in vitro dissolution. So we will see one by one. In that, general appearance, size, shape, and thickness. These are the involved in general appearance. So आपण general appearance का check करतो? This test is performed to facilitate packaging and to decide which tablet compressing machine which will be used. आपण टॅबलेटच्या साईज, शेप किंवा थिकनेस वरून डिसाइड करतो की आपण कोणत्या कम्प्रेशन मशीन यूज करायचे किंवा कोणत्या प्रकारचं पॅकेजिंग मटेरियल यूज करायचं टॅबलेट फॉर्म्युलेशन मध्ये. देन नेक्स्ट इज ऑर्गॅनोलॅक्टिक प्रॉपर्टीज. आपण ऑर्गॅनोलॅक्टिक प्रॉपर्टीज म्हणजे काय? विच टेस्ट विच आर परफॉर्म बाय युजिंग ऑर्गन्स. सो विच इन्क्लूड odor, color, taste of the tablet. Then next is thickness. Thickness can vary with no change in weight due to difference in density of granulation, the pressure applied to the tablet, the speed of the tablet compression. म्हणजे काय? टॅबलेटचा जो थिकनेस आहे तो कशा कशाने चेंज होतो, वेट आपण चेंज न करता टॅबलेटचा थिकनेस कशा कशाने चेंज होतो. सो ग्रॅन्युलेशनची जी डेन्सिटी आहे ती जर चेंज झाली तर थिकनेस व्हॅरी होतो. आपण टॅबलेट कंप्रेस करताना जे काही प्रेशर अप्लाय केले ते प्रेशर व्हॅरी असेल किंवा जे आपण प्रेशर अप्लाय केले त्याच्यानुसार थिकनेस चेंज होतो. देन नेक्स्ट इज द स्पीड ऑफ द टॅबलेट कंप्रेशन. टॅबलेट कंप्रेस करताना आपण स्पीड जास्त ठेवले की कमी ठेवले यानुसार द टॅबलेटची थिकनेस हा चेंज होतो. देन नेक्स्ट, टॅबलेट थिकनेस इम्पॉर्टंट इन रिप्रोड्युसिंग टॅबलेट्स identical in appearance but also to ensure that every production lot will be enabled or usable with selected packaging component. If the tablets are thicker than specified, a given number no longer may be contained in the volume of given size bottle. A plus or minus 5% may be allowed depending on the size of the tablet. So, thickness आपण कशाने मेजर करतो? वर्नियर कॅलिपरने. This diagram is of vernier caliper. Vernier caliperच्या मदतीने आपण thickness of tablet measure करतो. So next is hardness. Hardness is nothing but the crushing strength. It is the load required to crush the tablet when placed on the edge. Then why to perform measure hardness? आपण hardness test का perform करतो? To determine the need for pressure adjustment on the tablet machine. टॅबलेट मशीनवर प्रेशर किती अप्लाय केलं पाहिजे हे डिटरमाइन करण्यासाठी आपण हार्डनेस टेस्ट परफॉर्म करतो. टू विथस्टँड द मेकॅनिकल शॉक्स ऑफ मॅन्युफॅक्चरिंग, पॅकेजिंग अँड शिपिंग. आपण टॅबलेट जेव्हा मॅन्युफॅक्चरिंग प्रोसेस मध्ये असते तेव्हा टॅबलेट कितीतरी वेळा आपटली जाते, आदळली जाते किंवा त्याला शॉक्स बसतात पॅकेजिंग मध्ये किंवा शिपिंग मध्ये, मॅन्युफॅक्चरिंग मध्ये. सो ते किती टॉलरेट करू शकतात हे मेजर करण्यासाठी हार्डनेस चेक करतात. To ensure consumer acceptance. सपोज एखादी टॅबलेट खूप हार्ड असेल तर ती पेशंटच्या पोटात जाऊन किंवा ट्रॅक मध्ये जाऊन सुद्धा ती सोल्युबल होणार नाही. सो पेशंट एक्सेप्टन्स चेक करण्यासाठी आपण हार्डनेस चेक करतो. देन हार्डनेस कॅन अफेक्ट द डिसइंटिग्रेशन. So if the tablet is too hard, it may not disintegrate in the required period of time. And if the tablet is too soft, it will not withstand the handling during subsequent processing such as coating or packaging. म्हणजे काय?
हॅलो,
सॉरी सर. हा येस
काय मी करू का शेअर? सर मी करू का शेअर? काय होतं, प्रॉब्लेम येतोय का शेअरिंगला?
येस सर प्लीज. आय थिंक रेंज इश्यू आहे खूप.
ठीक आहे, तू फक्त बोल तिकडून, मी इकडे शेअर करतो. सर मला शेअर अलाव करा. वरती घेतलं नाही का? घे पडत डेस्कटॉपला, पडत घ्यायचा डेस्कटॉप.
अलाव केलं का? तर या ठिकाणी मॅडम आपल्याला टॅबलेटचे यूज आणि डिसएडव्हान्टेज ॲडव्हान्टेज माहिती देत आहेत. कुणाला जर काही प्रश्न असतील तर काढून ठेवा, मॅडम कडे द्या. तर या ठिकाणी मॅडम आपल्याला टॅबलेटचे यूज आणि डिसएडव्हान्टेज. तर आपल्याला माहिती आहे की आपल्या डेली यूज मध्ये टॅबलेटचं फार महत्व आहे. आपण अनेक प्रकारचे टॅबलेट घेत असतो आणि ते घेत असताना त्याचे ॲडव्हान्टेज काय, ते घेतल्या पाहिजेत का नाही, याची पण माहिती आपल्याला नसते. त्या दृष्टीने आपण कधी विचार करत नाही ते टॅबलेट मध्ये दिलेले कंटेंट कुठले आहेत, त्यासोबत जे काय आपल्याला घेतले जाणारे टॅबलेट्स योग्य प्रमाणात दिले जातात का नाही, याची सगळ्याची माहिती आपल्याला मॅडम शेअर करतायत. तोपर्यंत आपल्याला अनेक प्रश्न असतात त्या संदर्भात. असे जर काही प्रश्न असतील तर आपण कलेक्टिव्हली मॅडमशी शेअर करू. तसे प्रश्न असतील तर तुम्ही घेऊन काढा आणि त्यानंतर आपण ते मॅडमला विचारू आणि त्यांची उत्तर आपण घेण्याचा प्रयत्न करू.
हॅलो हॅलो
हा
सर.
बोला सर. हा सर.
स्लाइड शेअर करतोय मी.
हो
दिसते
सर.
का तुम्हाला? आता डेस्कटॉप दिसतोय बरोबर का माझा?
हो नाही सर, अजून दिसत नाही.
नाही, थांबा एक मिनिट,
एक
पुन्हा
हो.
हा पुन्हा करतो मी शेअर. एक मिनिट.
खडके सर जरा लक्ष द्या.
एक मिनिट हा. हा शेअर झाले का सर तुम्हाला?
नाही, माझ्याकडे अजून दिसत नाही. चौधरी
नाही
मला
का?
दिसतंय का?
थांबा एक मिनिट, एक मिनिट. दिसेल सर. सर आता शेअर झाली का बघा सर?
हो झाली
नाही
सर.
सर, अजून झाली का? ओके ओके.
आता शेअर झाली का बघा?
हा सर. हा
कोमल मॅडम.
हो हो
हा
हो
झाली सर तुमचे.
हो हो, जस्ट 1 मिनिट हा. बाकीचा
हा सर. हा झाली सर तुमची.
हो हो हो, मी थोडा वेळ फोन करतो, थोडा वेळ फोन करतो लेक्चर मध्ये. कोमल मॅडम. हॅलो.
येस सर.
हा, इथपर्यंत आला
हा
होता
सर.
तुम्ही? हा, प्लीज कंटिन्यू.
हार्डनेस.
आता नेक्स्ट और इट्स ओके?
नाही, इट्स ओके सर. नाही सर,
ओके
नेक्स्ट
प्लीज.
स्लाइड.
ओके.
Okay, so
और
hardness can affect disintegration. सपोज टॅबलेटचा हार्डनेस हा खूप असेल तर डिसइंटिग्रेशन मध्ये प्रॉब्लेम येतो. म्हणजे सपोज एखाद्या टॅबलेटला डिसइंटिग्रेट व्हायला 2 तास लागत असतील आणि टॅबलेट खूप हार्ड असेल तर ती टॅबलेट 2 तास होऊन गेले म्हणजे जो काही स्पेसिफाईड पिरियड असेल तरी ती टॅबलेट डिसइंटिग्रेट होत नाही. सो दिस टाइप ऑफ टॅबलेट इज रिजेक्टेड. And if the tablet is too soft, it will not withstand the handling due to subsequent processing, coating or packaging.
व्हॉइस का असा होते सर? मॅडम, चौधरी मॅडम. ओके प्लीज कंटिन्यू.
ओके, सपोज जर टॅबलेट ही खूप सॉफ्ट असेल तर त्या टॅबलेटला आपण कोटिंग करायला गेलो किंवा पॅकेजिंगच्या वेळी.
बोला सर.
नो नो, आवाज येत नव्हता व्यवस्थित म्हणून म्हणलं. ठीक आहे, आता ठीक आहे. माईक बंद करा, माईक बंद करा. ठीक आहे, कंटिन्यू मॅडम.
ओके, टॅबलेट जर खूपच सॉफ्ट असेल तर ती टॅबलेट ट्रॅव्हलिंगच्या वेळी किंवा पॅकेजिंगच्या वेळी ब्रेक होण्याचे चान्सेस खूप असतात. सो इन जनरल इफ द टॅबलेट हार्डनेस इज too high, we first check its disintegration before rejecting the patch, and if the disintegration is within limit, we accept the patch. म्हणजे काय तर टॅबलेटचा हार्डनेस हा खूप जास्त असेल तर पहिल्यांदा आपण डिसइंटिग्रेशन टेस्ट करतो. डिसइंटिग्रेशन टेस्ट केल्यानंतर स्पेसिफिक टॅबलेट ही डिसइंटिग्रेट होत असेल तर आपण ती जी काही बॅच असेल तो आपण ऍक्सेप्ट करतो किंवा अदरवाइज ती बॅच आपण रिजेक्ट करतो. सो सर नेक्स्ट स्लाइड.
ओके.
ओके, सो फॅक्टर्स अफेक्टिंग द हार्डनेस. कोणते फॅक्टर्स हार्डनेसवर अफेक्ट करतात? कंप्रेशन ऑफ टॅबलेट अँड कंप्रेसिव्ह फोर्स. सपोज जर फोर्स हा खूप जास्त झाला असेल तर टॅबलेट कंप्रेस होण्यावर अफेक्ट करतं. टॅबलेट ही खूप हार्ड होते किंवा कंप्रेशन फोर्स हा खूप कमी झाला असेल तर टॅबलेट ही खूप सॉफ्ट होते. अमाऊंट ऑफ बाइंडिंग एजंट आपण जर खूप जास्त टाकलं तर टॅबलेट हार्ड होते आणि बाइंडिंग एजंट सफिशियंट अमाउंट मध्ये जर आपण टाकली तर टॅबलेट ही प्रॉपर बनते. देन मेथड ऑफ ग्रॅन्युलेशन इन प्रिपेअरिंग द टॅबलेट, दॅट इज वेट ग्रॅन्युलेशन gives more hardness than direct method, and slugging method gives the best hardness. Slugging method ने जर आपण ग्रॅन्युल्स प्रिपेअर केले तर टॅबलेट ही प्रॉपर बनते. ओके, द लिमिट्स काय आहेत? ओरल टॅबलेटचा हार्डनेस किती असला पाहिजे? 4 to 10 kg हा ओरल टॅबलेटचा हार्डनेस असला पाहिजे. But in hypodermic tablets and chewable tablets, hardness is 3 kg, and in sustained release tablets, hardness is 10 to 20 kg. सस्टेन रिलीज मध्ये हार्डनेस हा इतका जास्त का असतो? तर सस्टेन रिलीज टॅबलेट म्हणजे काय असतात? त्या आपल्या बॉडीमध्ये मोर टाइम साठी असतात. म्हणजे त्यांचा जो काही पिरियड आहे तो जास्त असतो आपल्या बॉडीमध्ये डिझॉल्व्ह होण्याचा किंवा ऍब्झॉर्ब होण्याचा. सो त्या टॅबलेटचा हार्डनेस आहे तो 10 to 20 kg असतो. सर नेक्स्ट स्लाइड.
ओके, नेक्स्ट दिसते का स्लाइड?
हो सर. सो
ओके.
आपण हार्डनेस परफॉर्म करतो. आपण मगाशी बघितलं जो हार्डनेस होता तो हार्डनेस टेस्टर होता. त्या टेस्टर मध्ये आपण 5 टॅबलेटचा हार्डनेस चेक करतो आणि त्यांचा ऍव्हरेज काढतो आणि त्यानुसार आपण हार्डनेस चेक करतो. देन नेक्स्ट इज फ्रायबिलिटी. फ्रायबिलिटी इज द टेंडन्सी ऑफ टॅबलेट्स टू पावडर, चिप और फ्रॅगमेंट, and this can affect the appearance, consumer acceptance of the tablet, and also add to tablet's weight variation or content uniformity problems. Friability टेस्ट म्हणजे काय? आपण टॅबलेटची टेंडन्सी चेक करतो की ट्रॅव्हलिंग मध्ये किंवा पॅकेजिंग मध्ये टॅबलेटची पावडर किती लॉस होतीये किंवा त्यामध्ये काही क्रॅक्स येत आहेत का, चिप्स येत आहेत का, किंवा ट्रॅव्हलिंग मध्ये सपोज टॅबलेटची पावडर होतात किंवा क्रॅक्स केले तर त्याचा एलिगन्स जो अपिअरन्स आहे तो चेंज होतो. त्यामुळे पेशंट ऍक्सेप्टन्स हा म्हणजे एक्झाम्पल घ्या, आपण सपोज एखादी टॅबलेट ब्रेक झालेली तर आपण खाऊ का? आपण घेण्याआधीच टॅबलेट ऑलरेडी ब्रेक झाली आहे. सो दॅट अफेक्ट ऑन टॅबलेट अँड ऑल्सो ऍड टू टॅबलेट वेट व्हारिएशन अँड कंटेंट युनिफॉर्मिटी प्रॉब्लेम्स. म्हणजे ट्रॅव्हलिंग मध्ये टॅबलेट जर लॉस होत असेल ड्रग तर ते वेट व्हारिएशन मध्ये आणि कंटेंट युनिफॉर्मिटी मध्ये प्रॉब्लेम्स क्रिएट करतं. सो नेक्स्ट. So friability is a property that is related to the hardness of the tablet. An instrument called friablator is used to evaluate the ability of the tablet to withstand abrasion in packaging, handling, and shipping. Friablator determines friability to allowing tablet to roll and fall 6 inches within the rotating tumbling apparatus. This apparatus is friablator. आपण ह्या फ्रायब्लेटर मध्ये फ्रायबिलिटी चेक करतो. सो प्रोसिजर काय आहे फ्रायबिलिटीची? 20 टॅब्लेट्स घ्यायच्यात एकत्र, त्या टॅब्लेट्स वेट करायच्या. Put this tablet in friablator and adjust the instrument at 100 rpm. त्या टॅबलेट घ्यायच्या, वेट करायच्या, पहिल्यांदा त्या फ्रायब्लेटर मध्ये ऍड करायच्या, फ्रायब्लेटर मध्ये प्लेस करायचा आणि जो काही प्रोग्राम असेल, इन्स्ट्रुमेंट असेल ते सेट करायचं 100 rpms ला, म्हणजे 25 rpms फॉर 4 मिनिट्स, म्हणजे 25 rpm 1 मिनिट साठी सेट करायच्या. 4 मिनिट मध्ये 100 rpm होतात. The next step is wait the 20 tablets. म्हणजे after 4 minutes wait these 20 tablets. Then how to calculate? Formula काय आहे त्याचा? Friability is equal to 100 into 1 minus W by W. So W काय? Weight of tablets before friability. Friability करण्याच्या आधीचं weight, and W म्हणजे काय? Weight of tablets after friability. Okay. Friability is equal to then जे काही येईल ते वेट, इट मस्ट बी लेस दॅन और इक्वल टू 1% ऑफ ड्रग. म्हणजे सपोज जी काही टॅबलेट असेल त्यामधून 1% किंवा 1% पेक्षा कमी जर ड्रग लॉस झालेला असेल किंवा कंटेंट लॉस झाला असेल तर ती टॅबलेट किंवा ती बॅच इज ओके, और अदर दॅन विच रिजेक्ट दिस बॅच. सम चिवेबल टॅबलेट्स अँड मोस्ट इफरसन टॅबलेट्स आर हायली फ्रायबल अँड रिक्वायर स्पेशल युनिट पॅकेजिंग. म्हणजे ज्या काही चिवेबल टॅबलेट्स असतात किंवा मोस्ट इफरसन टॅबलेट्स असतात त्या ऍज कम्पेअर टू अदर टॅबलेट्स सॉफ्ट असतात. सो त्यांच्यासाठी स्पेशल पॅकेजिंग मटेरियल किंवा स्पेशल पॅकेजिंग यूज केलं जातं. सो नेक्स्ट is weight variation. Variation is nothing but uniformity of weight of the tablets. The weight variation test would be satisfactory method for determining drug content uniformity of drug distribution. Weight variation test is applicable when the tablets containing 50 mg or more of drug substance, or when the drug substance represents 50% or more by weight of the dosage unit form. म्हणजे टॅबलेट मध्ये 50 mg किंवा 50 mg पेक्षा जास्त ड्रग असेल, म्हणजे फक्त ऍक्टिव्ह फार्मास्युटिकल इन्ग्रेडियंट हा 50 mg किंवा 50 mg पेक्षा जास्त असेल तर आपण ही टेस्ट परफॉर्म करतो. किंवा ओके, ही टेस्ट आपण कशी परफॉर्म करतो? वेट 20 टॅबलेट्स सिलेक्टेड ऍट रॅन्डम, इच वन इंडिव्हिज्युअली. म्हणजे 20 टॅबलेट्स घ्यायच्यात, त्या 20 टॅबलेट्स वेट करायच्यात इंडिव्हिज्युअली. ओके, आणि नंतर इंडिव्हिज्युअली वेट केल्याच्या नंतर डिवायडेड बाय 20 टॅबलेट्स घेतल्यामुळे डिवायडेड बाय 20. सर नेक्स्ट स्लाइड. अ 20 टॅबलेट्स घेतल्यानंतर त्यांचं वेट करायचंय, 20 टॅबलेटचं वेट घ्यायचंय डिवायडेड बाय 20, ऍव्हरेज वेट काढायचंय आणि प्रत्येक टॅबलेट आणि ऍव्हरेज वेट ह्यांच्यामध्ये कंपॅरिझन करायचे. आणि त्यामध्ये 2 टॅबलेट्स किंवा 2 पेक्षा जास्त टॅबलेट जर ऍव्हरेज वेट पेक्षा डिफरन्स असेल, प्लस ऑर मायनस 5 ने डिफरन्स असेल तर आपण ती बॅच रिजेक्ट करतो. आणि 2 टॅबलेट्स पेक्षा कमी म्हणजे 1 किंवा 2 टॅबलेट्स असतील तर इट्स ओके, बट 3, 4, 5 अशा टॅबलेट्स जर व्हॅरी होत असतील तर आपण ती बॅच रिजेक्ट करतो. ओके, नेक्स्ट इज कंटेंट युनिफॉर्मिटी टेस्ट. कंटेंट युनिफॉर्मिटी टेस्ट कशी परफॉर्म करायची आहे? रॅन्डमली सिलेक्ट 30 टॅबलेट्स, 10 ऑफ दीज ऍसेड इंडिव्हिज्युअली. त्या 30 टॅबलेट्स मधल्या 10 टॅबलेट्स आहेत त्या ऍसेड करायच्यात इंडिव्हिज्युअली. ऍसे म्हणजे काय? आपण बघतो की एक 10 टॅबलेट्स घ्यायच्यात, 1 टॅबलेट घ्यायची, ट्रायट्रिएट करायची आणि मग त्याचा ऍसे करायचा आणि नंतर UV ला चेक करायचं. देन द टॅबलेट्स पास द टेस्ट इफ 9 of the 10 tablets must contain not less than 85% and not more than 115% of the labeled drug content. ओके, त्या 10 टॅबलेट मधल्या 9 टॅबलेट हा 85% पेक्षा कमी नसल्या पाहिजे आणि 115 पेक्षा जास्त ड्रग लॉस झालेला नसला पाहिजे. देन 10 टॅबलेट मे नॉट कंटेन जी 10वी टॅबलेट आहे मे नॉट कंटेन 75%, less than 75% and more than 125% of the labeled content. Then if the conditions are not met, remaining 20 tablets acid. जर ह्या कंडिशन मध्ये टॅबलेट ऍसे बसत नसेल तर आपल्याला रिमेनिंग 20 टॅबलेट्स आहेत त्याचा परत ऍसे करायचा आहे अँड नॉन मे फॉल आउटसाइड ऑफ द 85% to 115% range. It is required for all coated and uncoated tablets containing less than 50 mg of an active ingredient. Comprising less than 50% of weight of one dosage unit. 9 of 10 must contain labeled amount plus or minus 15% and non exceed 25%. Next test is disintegration test. It is the time required for the tablet to break into particles. The disintegration test is measure only of the time required under the given set of conditions for a group of tablets to disintegrate into particles which will pass through 10 mesh screen. Okay. So disintegration test म्हणजे काय? Time required for the tablet to break into particles. टॅबलेटला पार्टिकल्स मध्ये ब्रेक व्हायला किती टाइम लागतो हे त्या टेस्ट मध्ये मेजर केलं जातं. सो disintegration test is measure only of the time required under the given condition for the group of tablets. हे ग्रुप मध्ये आपण परफॉर्म केलं जातं. इंडिव्हिज्युअली टेस्ट ही परफॉर्म केली जात नाही. टू डिसइंटिग्रेट इनटू पार्टिकल्स विच विल पास थ्रू 10 मेश स्क्रीन. 10 मेश स्क्रीन नी पास केली जाते. So disintegration apparatus consists. The next slide. The disintegration apparatus consists of basket rack. आपण बघू शकतो basket rack असते ह्या apparatus मध्ये holding 6 plastic tubes. So disintegration apparatus मध्ये basket rack होल्ड केले जातात. त्यामध्ये 6 प्लास्टिकच्या ट्यूब असतात. ओपन ऍट टॉप अँड बॉटम. त्या दोन्हीकडे टॉपला आणि बॉटमला ओपन असतात. द बॉटम ऑफ द ट्यूब्स इज कवर्ड विथ 10 मेश स्क्रीन. जे काही ट्यूब्सचं बॉटम आहे ते 10 मेश स्क्रीनने कव्हर केलेलं असतं. द बास्केट रॅक इज इमर्स्ड. ते बास्केट रॅक इज इमर्स्ड इन अ बाथ ऑफ सुटेबल लिक्विड. देन आपण इथं बघू शकतोय जे काही सुटेबल लिक्विड असेल त्यामध्ये ते जे रॅक आहे ते आपण डीप करतो और इमर्स करतो. हेल्ड ऍट 37 डिग्री सेल्सिअस. त्याचं टेम्परेचर हे मेंटेन केलं असतं 37 डिग्री सेल्सिअस. प्रेफरेबली इन 1 लिटर बीकर. The rack moves up and down in the fluid at a specific rate. ते जे काही 6 ट्यूब होल्ड केलेलं रॅक आहे ते अप अँड डाऊन मूव्ह होतं at specific rate. For compressed uncoated tablets, the testing fluid is जे काही 6 ट्यूब होल्ड केलेलं रॅक आहे ते अप अँड डाऊन मूव्ह होतं at specific. Just uncoated tablets, the testing fluid is up and down degree Celsius. But in some cases, the monographs direct that. Some cases मध्ये मोनोग्राफ काय डायरेक्ट करतात. सम केसेस मध्ये आपण काय यूज करतो सिम्युलेटेड गॅस्ट्रिक फ्लुइड आपण यूज करतो. सो ऍपरेटस आपण बघू शकतोय. देन नेक्स्ट आपण कोणकोणती लिक्विड्स यूज करतो डिसइंटिग्रेशन मध्ये. वॉटर, सिम्युलेटेड गॅस्ट्रिक फ्लुइड अँड सिम्युलेटेड इंटेस्टिनल फ्लुइड. आपण वॉटर हे अनकोटेड टॅबलेट साठी मोस्टली यूज केलं जातं आणि सिम्युलेटेड गॅस्ट्रिक फ्लुइड आणि इंटेस्टिनल फ्लुइड हे दोन वेगवेगळे का करतो तर स्टमकचा पीएच आणि इंटेस्टिनलचा पीएच हा वेगवेगळा असतो. सो आपण गॅस्ट्रिक पीएच असणारं पण फ्लुइड यूज करतो आणि इंटेस्टिनल पीएच असणारं फ्लुइड सुद्धा आपण यूज करतो. आणि आपण बघू शकतो इथे ही त्या ट्यूब्स आहेत. देन दिस इज द प्रोसेस ऑफ डिसइंटिग्रेशन. For most uncoated tablets, the period is not more than 30 minutes. According to USP. USP नुसार ज्या uncoated tablets आहेत त्याला पिरियड किती लागतो? 30 मिनिट पेक्षा जास्त वेळ नाही लागत डिसइंटिग्रेट व्हायला. अकॉर्डिंग टू ब्रिटिश फार्माकोपिया, अनकोटेड टॅबलेटला 15 मिनिट्स लागतात ब्रेक व्हायला. Although the time for uncoated tablet varies greatly. From this, for coated tablets, up to 2 hours may be required. ज्या काही coated tablets असतील त्यांना 2 तास लागतात. While for sublingual tablets, disintegration time is 3 minutes. Sublingual tablets हा आपण orally administer करतो किंवा आपण त्या administer न करता सबलिंगुअली आपण ऍडमिनिस्टर करतो. सो त्याचा जो काही डिसइंटिग्रेशन टाइम आहे तो कमी आहे, कारण त्या इझिली ब्रेक झाल्या पाहिजे. देन द टॅबलेट डिसइंटिग्रेशन टेस्ट इज लिमिटेड टू मॅन्युफॅक्चरिंग कंट्रोल ऑफ लॉट टू लॉट व्हेरिएशन इन इंडिव्हिज्युअल प्रोडक्ट अँड इट इज नॉट मेजर ऑफ बायोअबिलिटी. आपण डिसइंटिग्रेशन टेस्ट नुसार बायोअबिलिटी हे डिटेक्ट नाही करत किंवा प्रेडिक्ट नाही करत. इट इज यूज टू प्रोव्हाइड अ सिम्पल अँड युजफुल मीन्स ऑफ मॉनिटरिंग अँड कंट्रोलिंग द क्वालिटी ऑफ द टॅबलेट. आपण डिसइंटिग्रेशन टेस्ट नुसार बायोअबिलिटी ही डिटेक्ट प्रेडिक्ट नाही करत, बट दिस इज कंट्रोल द क्वालिटी ऑफ द टॅबलेट. त्यावरून आपल्याला टॅबलेटची क्वालिटी ही समजते. देन नेक्स्ट, USP मेथड फॉर अनकोटेड टॅबलेट्स. अनकोटेड टॅबलेट्स आपण परफॉर्म कसं करतो? स्टार्ट द डिसइंटिग्रेशन टेस्ट ऑन 6 टॅबलेट्स. 6 टॅबलेट्स त्यामध्ये फिल करून disintegration test start करायचे. If 1 or 2 tablets from the 6 tablets fail disintegrate completely within 30 minutes, repeat the same test on another 12 tablets. If the whole test will consume 18 tablets, not less than 16 tablets disintegrate completely within the time. ज्या काही टॅबलेट्स आपण परफॉर्म केलेले असतील त्यामधून 2 किंवा 2 पेक्षा जास्त टॅबलेट जर फेल जात असतील तर ती टेस्ट is not passed. If the more than 2 tablets from the 18 fail to disintegrate, the batch must be rejected. जर 18 मधील जर 2 पेक्षा जास्त टॅबलेट्स ह्या फेल होत असतील डिसइंटिग्रेट व्हायला तर बॅच मस्ट बी रिजेक्टेड. देन लिमिट्स काय आहेत अनकोटेड टॅबलेट्सचे? अकॉर्डिंग टू USP, मीडियम आपण यूज करतो सिम्युलेटेड गॅस्ट्रिक फ्लुइड. त्यासाठी टेम्परेचर मेंटेन केलेलं असतं 37 डिग्री सेल्सिअस आणि टाइम इज नॉट एक्सीड दॅन 30 मिनिट्स. अकॉर्डिंग टू ब्रिटिश फार्माकोपिया, आपण मीडियम हे वॉटर यूज करतो. टेम्परेचर हे 37 डिग्री सेल्सिअस असतं आणि टाइम पिरियड इज नॉट एक्सीड दॅन 15 मिनिट्स. Next test is dissolution test. So dissolution test is the process by which a solid enters in a solution. म्हणजे dissolution test काय आहे? जे काय आपले टॅबलेट असेल तर सोल्युशन मध्ये पूर्णपणे मिक्स होते, त्याला dissolution test असं आपण म्हणतो. Dissolution rate is defined as the amount of drug substance that goes into solution per time under the standardized condition of liquid and solid interference, temperature and solvent composition. Dissolution is one of the most important quality control tests and considered as a tool for predicting bioavailability in some cases, replacing clinical studies to determine bioequivalence. In fact, a direct relationship between in vitro dissolution rate of many drugs and their bioavailability has been demonstrated and is generally referred as in vitro and in vivo correlation. डिझोल्युशन टेस्ट मध्ये आपण काय करतो? परफॉर्म कशी करतो आपण डिझोल्युशन टेस्ट? आपलं जी काही बॅच परफॉर्म केलेली असतील त्यामधील टॅबलेट घेतो. ती टॅबलेट आपण मोस्टली आपण टॅबलेट साठी कोणतं ऍपरेटस यूज करतो? टाइप सेकंड वाले टॅबलेट पॅडल वाले जे ऍपरेटस असतं ते आपण यूज करतो. आणि नेक्स्ट स्लाइड. Variety of designs of apparatus for dissolution testing is varying simple beaker to complex system, where an attempt to made to mimic the biological media. The choice of the apparatus to be used depends largely on the physiochemical properties of the dosage form. Apparatus आपण physiochemical properties of dosage form टॅबलेटच्या ज्या काही physiochemical properties असतात किंवा जो काय आपला dosage form असेल त्याच्या प्रॉपर्टीज नुसार आपण टॅबलेट्सचं ऍपरेटस हे डिसाईड करतो किंवा डिझोल्युशनचं ऍपरेटस डिसाईड करतो. डिझोल्युशन ऍपरेटस. तर डिझोल्युशन ऍपरेटस किती प्रकारचे असतात? 8-9 प्रकारचे डिझोल्युशन ऍपरेटस असतात. इन दॅट फर्स्ट इज रोटेटिंग बास्केट. रोटेटिंग बास्केट मध्ये आपण कोणकोणते डोसेज फॉर्म परफॉर्म करतो? टॅबलेट्स. देन सेकंड वन इज पॅडल. Paddle is mostly used apparatus for dissolution. In that we perform tablet, capsule, modified drug products and suspension. Third one is reciprocating cylinder. Reciprocating cylinder मध्ये आपण कोणकोणत्या टॅबलेट किंवा डोसेज परफॉर्म करतो? एक्सटेंडेड रिलीज जे काय असतील टॅबलेट्स ते आपण रेसिप्रोकेटिंग सिलेंडर मध्ये परफॉर्म करतो. देन नेक्स्ट इज फोर्थ वन इज फ्लो सेल. फ्लो सेल मध्ये आपण कसले ड्रग किंवा डोसेज फॉर्म परफॉर्म करतो? ड्रग प्रोडक्ट कंटेनिंग लो वॉटर सोल्युबल ड्रग्स. ज्या काही जे लो वॉटर सोल्युबल ड्रग्स असतील ते आपण फ्लो सेल मध्ये परफॉर्म करतो. नेक्स्ट इज पॅडल ओव्हर डिस्क. पॅडल ओव्हर डिस्क मध्ये आपण कसले डोसेज फॉर्म परफॉर्म करतो? ट्रान्सडर्मल प्रोडक्ट. नेक्स्ट इज सिलेंडर. सिलेंडर मध्ये ट्रान्सडर्मल ड्रग प्रोडक्ट्स अँड reciprocating disc, also transdermal drug products. Then next is non-USPNF. In that extended release drug products and non-USPNF. In that ointment, creams, transdermal drug products. So next, this is type 1 apparatus, rotating basket. आपण बघू शकतो इथं फर्स्ट जे काही बास्केट आहे त्यामध्ये आपण डोसेज फॉर्म फिल केलेला असतो आणि देन आपण ऍपरेटस स्टार्ट करतो. देन रोटेटिंग बास्केट काय आहे? इन केस ऑफ नॉन डिसइंटिग्रेटिंग डोसेज फॉर्म दिस ऍपरेटस इज सुपेरियर टू ऍपरेटस 2 since it constrains the dosage form in the steady state fluid flow. It is inferior for testing dosage forms which contains gums due to clogging of screen matrix. Okay. So next is this is rotating paddle. This is type apparatus 2. Mostly आपण टॅबलेट साठी हे सेकंड वाला apparatus यूज करतो, रोटेटिंग पॅडल. आपण बघू शकतो टॅबलेट ही एकदम बॉटमला आहे. So in rotating paddle, this apparatus is identical to apparatus 1 except that the paddle is substituted for the rotating basket. Frequently used for both disintegrating and non-disintegrating dosage forms. This diagram is of reciprocating cylinder. Okay. So reciprocating cylinder काय आहे? One advantage of reciprocating cylinder is that the gastrointestinal tract conditions can be easily stimulated as it is easy to make time dependent pH changes. Reciprocating cylinder मध्ये आपण गॅस्ट्रोइंटेस्टिनल जो काही pH आहे तो आपण इझिली मेंटेन करू शकतो. सो त्या टाइपचे जे ड्रग्स असतील ते आपण त्यामध्ये परफॉर्म करतो. दिस ऍपरेटस इज मोस्ट सुटेबल फॉर नॉन डिसइंटिग्रेटिंग that is the extended release or delayed release that is enteric coated dosage form. हे apparatus कशासाठी सुटेबल आहे? एक्सटेंडेड रिलीज आणि enteric coated tablets साठी हे apparatus सुटेबल आहे. Then next is flow cell. That's the fourth apparatus is flow cell. The advantage of flow through cell apparatus is the ability to test drugs of very low aqueous solubility. ज्या ड्रग्सची सोल्युबिलिटी ही ज्या ड्रग्सची लो aqueous solubility ते आपण त्यामध्ये परफॉर्म करतो and the ability to change the pH conveniently during the test. ओके, जे ड्रग pH चेंज करतात ड्युरिंग टेस्ट त्या ड्रग साठी आपण हे ऍपरेटस यूज करतो. सो नेक्स्ट इज पॅडल ओव्हर डिस्क. म्हणजे आपण बघितलं सेकंड वाला पॅडल सारखं होतं. सो बट त्यामध्ये डिस्क नव्हती. ह्या ऍपरेटस हे मॉडिफाईड आहे पॅडलचं. त्यामध्ये डिस्क ही अप्लाय केलेली आहे. इन दॅट द नेक्स्ट इज सिलेंडर. सिक्स्थ ऍपरेटस इज सिलेंडर. द सिलेंडर मेथड for testing transdermal preparation is modified from the basket method, that is apparatus 1. ही apparatus 1 च modified method आहे. apparatus 6. In place of basket, a stainless steel cylinder is used to hold the sample. basket मध्ये stainless steel च cylinder यूज केलेले आहे सॅम्पल होल्ड करण्यासाठी. Next is reciprocating disc method. Reciprocating disc method काय आहे? In the reciprocating disc method for testing transdermal products, a motor drive assembly is used to reciprocate the system vertically, and the samples are placed on the disc-shaped holders using Cufron supports. Okay. Next is all 6 tablets must meet the requirements specific. If one or two tablets fail, repeat the test on 6 additional tablets. In most cases, the amount of drug dissolved should not be less than 70% of quantity contained in the tablets after 45 minutes. The formulation and manufacturing factors affecting the dissolution of tablet. The particle size of the drug substance. ज्या काही particle size ड्रग्सची particle size आहे ते affect करते dissolution वर. The solubility and hygroscopicity of the formulation. ड्रगची solubility and hygroscopy also affect on the dissolution of tablet. The type of concentration of the disintegrant, binder and lubricant can used. The manufacturing method, particularly the compactness of granulation and the compression force. Granulation मधला compactness आणि compression force is also affect on the dissolution rate. Various pharmacopoeias contains specifications on dissolution, requirement of various drugs. That is, monograph specifies stirring speed, temperature, viscosity, pH, composition of dissolution media, and presence or absence of wetting agents. These these contents also affect on the dissolution of tablet. Okay, thank you.
Thank you, madam. So this session is very, very informative as such. So there are some questions, ma'am, for you from our student. Please, ma'am.
येस मॅम. तर काय टॅबलेटला कव्हरिंग असते ते कव्हरिंग काढून खाल्लं तर चालते का, किंवा ते म्हणजे कव्हरिंग कशासाठी असते नेमकं?
सपोज एखाद्या टॅबलेट मधील जो ऍक्टिव्ह इन्ग्रेडियंट आहे तो खूप बिटर असेल किंवा त्याचा अनप्लेझंट टेस्ट किंवा फ्लेवर असेल, तो आपण हाईड करण्यासाठी कोटिंग वापरतो. किंवा असेल ड्रग, एखादी टॅबलेट असेल तर ते लूज होण्यासाठी आपण टॅबलेट.
हॅलो.
किंवा जेडियाट्रिक पेशंट मध्ये सपोज अनप्लेझंट टेस्ट असेल तर पेशंट खात नाही टॅबलेट, तर आपण शुगर कोटिंगने टॅबलेट देतो. सो टेस्ट ही रिमूव्ह करण्यासाठी किंवा मास्क करण्यासाठी आपण कोटिंग करतो.
ठीक आहे मॅम. आणखीन एक क्वेश्चन होतं. वेगवेगळ्या टॅबलेटला वेगवेगळे कलर पाहायला मिळतात.
आवाजच नाही.
तर आवाज सर क्लिअर आहे ना?
हा, आता आवाज येतो का?
हा, बोला सर.
हा, तर काही टॅबलेट कलर वेगवेगळे असतात, तर ते त्याचं रिझन काय त्यामागे?
बेस्ड ऑन फिजिओकेमिकल प्रॉपर्टीज. तर कलर मध्ये सुद्धा फिजिओकेमिकल प्रॉपर्टीज असतात, त्या आपल्या ड्रगशी कंटॅमिनेटेड नाही झाल्या पाहिजे, किंवा दोन्ही मध्ये कंपॅटिबल जो कलर असेल कंपॅटिबिलिटी चेक करून हे ड्रग मध्ये कलर किंवा जे काही एजंट्स असतात ते आपण युज करतो कंपॅटिबिलिटीनुसार. म्हणजे जो कंपॅटिबल असेल तो आपण युज करतो टॅबलेटला.
हॅलो.
ठीक आहे मॅम. तर ते पॅरासिटामॉल टॅबलेट आणि औषध मध्ये काय फरक आहे?
कशाची?
पॅरासिटामॉल टॅबलेट
आणि औषध, सिरप आणि
टॅबलेट. तसं बघितलं तर फरक त्याचं जे काम आहे ते काम सेमच आहे. बट काही पेशंट मध्ये पेशंट टॅबलेट घेत नाही, म्हणजे सॉलो करत नाही. सो आपण त्यांना थ्रू देऊ शकतो. म्हणजे ऍडमिनिस्टर करण्यासाठी आपण इझी कोणतं आहे त्यानुसार आपण हे सिरप आणि टॅबलेट देतो. फरक असा त्याचं दोन्हीचं काम एकच आहे.
हॅलो.
येस सर.
हॅलो सर, जसं की लहान मुलाला सिरप दिला जातो, लहान मुलं बाळ तर टॅबलेट घेऊ शकत नाही ना. हॅलो, आवाज येतो का सर?
पेशंटच्या कन्व्हिनियन्स आपण
म्हणजे लहान मुलं टॅबलेट खात नसल्यामुळे आपण सिरप देतो.
हा, सिरप देतो. आणि एकदम म्हातारी माणसं ज्याला गिळणं शक्य नाही किंवा पेशंटला सॉल्व्हिंग करणं टॅबलेट शक्य नाही, ह्याला सुद्धा सिरप
डिपेंडेड अपॉन दॅट पेशंट.
हा, पेशंट कॉम्प्लेक्स.
Okay. Okay, sir. Thank you, ma'am. Thank you, Khadar ma'am, for
सर
focusing. हाँ,
सर एक मिनिट सर सर अजून एक
हाँ.
क्वेश्चन आहे मुलाला
हा बोला बोला.
एक मिनिट सर एक क्वेश्चन घ्या सर अजून
हा सांगा.
What happen when a batch fails quality control test?
ओके. पेन कोमल कॅन आय आन्सर?
सपोज जर
ओके.
बॅच
आवाज काय येतो सर? दुसरा एक आवाज येतो. हा बोला. हा
सपोज जर एखाद्या टॅबलेटची बॅच फेल गेली क्वालिटी कंट्रोल टेस्ट मध्ये तर आपण ऑब्विसली ती रिजेक्ट करतो. ती कशामुळे फेल गेली सपोज आपण हार्डनेसची टेस्ट घेऊयात. हार्डनेस ती खूप जास्त हार्ड झाली आहे तर ती का झाली आहे त्यामागची रिझन बघतो आणि ती आपण करून पुन्हा बॅच बनवतो. जी काही फेल गेलेली बॅच आहे ती फेल असते. ती मॅन्युफॅक्चरिंग मध्ये आलेली नसते किंवा मार्केटमध्ये आलेली नसते. सो आपण ती तिथंच रिजेक्ट करून त्यामध्ये इम्प्रुव्हमेंट करतो.
ओके. मी अजूनही सांगू शकतो. कोमल एक मिनिट. हा सर आवाज येतो माझा?
हो सर, हो सर.
जर एखादी बॅच फेल गेली समजा पास झाली नाही, पास नाही बॅच, जी बॅचला ओके असते तिला आपण पास लेबलिंग असते इंडस्ट्रीमध्ये बरं का, आणि जी फेल जाते म्हणजे जे रिजेक्ट होती रिजेक्ट नाही तिला फेल म्हणलं जातं. आता फेल गेलेल्या बॅचचं पुन्हा रिपीट क्वालिटी कंट्रोल केलं जातं आणि पुन्हा दुसऱ्या वेळेस फेल गेली तर मग ती बॅच डिस्पॅच होत नाही. जर दुसऱ्या वेळेस ओके चालली, कारण देअर मे बी द मॅन मिस्टेक, ह्युमन मिस्टेक पण कधी कधी होऊ शकते बॅच म्हणजे प्रॅक्टिकल करताना, क्वालिटी कंट्रोल टेस्ट करताना, आणि मग दुसऱ्या वेळेस ती फेल गेली तर मग त्याचे रिझन शोधले जातात आणि ती बॅच बॅन केली जाते. असं आहे. कशातही असू द्या, म्हणजे नुसतं हार्डनेस मध्ये नाही, सगळ्या कुठल्याही प्रकारामध्ये लगेच कंपनी रिजेक्ट करत नाही तर दोनदा स्टडी केला जातो आणि मग त्यानंतर दोन्ही केसमध्ये सेम रिझल्ट आले तर बॅच विल बी रिजेक्टेड, दॅट इज फेल, अँड फायनली इट विल बी रिजेक्टेड.
ओके, ओके. थँक्यू सर.
एनी क्वेश्चन?
Okay, no, sir. आता मी हे करतो vote of thanks. So, thank you, madam. This session is very informative, and we got very much new information about the tablets, their uses, and so more. So, madam focuses on quality control test for tablets, and thank you, ma'am, for that fascinating presentation. Your speech is truly inspiring, and I hope you all present here have gained valuable insights into the future use of the tablets. A big thank you to our esteemed speakers, Khata ma'am, for sharing their expertise. We also extend our sincere gratitude to our Microbiology Society PM Usha scheme for their generous support. And finally, thank you to all of you for your active participation. Thank you one and all.
Okay. Thank you, sir.
Oh, thank you, sir. Chaudhary madam.
Hello.
Thank you, sir.
Okay.
The session is over. So now I request Chaudhary madam, please declare the next schedule of the workshop.
सर्व विद्यार्थ्यांना एक सूचना आहे. हा सेशन एंड झालेला आहे आणि यानंतरचा जो सेशन आहे तो एक्झॅक्टली 2 वाजता सुरू होईल. सगळ्यांनी YouTube लाईव्ह वरती 2 वाजता शार्प प्रेझेंट राहायचंय. लंच ब्रेक आहे.
ओके, थँक्यू सर.
सर्व विद्यार्थ्यांनी 2 वाजता जॉईन करा YouTube लाईव्ह वरती. लंच ब्रेक आहे आता 1 ते 2.
चालू कर मग मला मी करतो मी.
Welcome, madam.
Welcome and good afternoon all. Just let me know, madam, when can we start our
Hello.
session?
चौधरी मॅडम कॅन आय सर?
हाँ. सर स्टार्ट कर
गुड
दे.
आफ्टरनून.
Yes, sir. Start करा.
Good afternoon, everyone. Myself, Dr. Lakshman, Assistant Professor in Venkatesh Mahajan Senior College, Dharashaw. Here we will start the third session of the today's workshop. It's my pleasure to introduce our today's speaker, Dr. Sheetal Chaitanya Pednekar, madam, who is currently working as an Associate Director at Biocon Biologics Limited in Bangalore. His qualification is M Pharma with PhD. Dr. Pednekar, madam, is an expert in the biopharmaceutical industry with extensive experience in regulatory affairs,
सर तुमचा
compliance.
साऊंड व्यवस्थित येत नाही सर.
Yes.
सर जरा फास्ट होत आहे आणि साऊंड व्यवस्थित येत नाही.
Okay. Dr. Pednekar, madam, uh, is currently working as Associate Director at Biocon.
सर 1-2 मिनिट थांबा सर. फास्ट होत आहे. 1-2
Okay.
मिनिट थांबा सर.
Okay, ma'am. Okay.
साऊंड रीच होत नाही इथं तुमचं विद्यार्थ्यापर्यंत.
Okay.
कोण दिले विद्यार्थ्यांना लिंक रे? कुणी विद्यार्थी जॉईन होईल? नाही सरांनी दिली नाही. अलाउड नाही करत. सॉरी फॉर इंटरप्शन. जरा छोटासा टेक्निकल प्रॉब्लेममुळे तर आवाज रीच होत नाही आहे मॅडम. एक
No
जस्ट
problem,
स्टार्ट
madam.
करूया
Take your
आपण.
time.
ओके.
No problem.
मध्ये यायला सांगा. Clear, sir? Sir. Sir, you can continue, sir. Clear, sir? Introduction continue करा. Madam. Sir, you can continue, sir.
Madam.
Introduction continue करा.
Okay, madam. Here we will start the third session of the today's workshop. It's my pleasure to introduce our speaker, Dr. Sheetal Chaitanya Pednekar, madam. She is currently working as an associate director at Biocon Biologics Limited in Bangalore. His qualification is M Pharma PhD. Dr. Pednekar, madam, is an expert in the biopharmaceutical industry with extensive experience in regulatory affairs, compliance, and the production of biological products. Her deep understanding of the complex regulatory landscape governing biologics has been instrumental in ensuring compliance with global regulatory agencies, including the FDA, EMA, and CDSCO. Today, she will be sharing her insights on regulatory requirements of biological product production, a critical topic in the field of the biopharmaceuticals. Dr. Pendigar, madam, will be provide us with valuable perspectives on the key regulatory frameworks, quality standards, and best practices necessary for ensuring the safety, efficacy, and quality of biological products. Please join me in welcoming Dr. Sheetal Chaitanya Pendigar, madam. Please go ahead, madam.
Yeah. Thank you, Dr. Chitade, sir. And I really thank to Venkatesh Mahajan College for Pharmacy who has conducted this workshop or online uh, discussion or forum for students in Pharmacy College. Uh, my basic topic is with respect to the product approval in biological. What is the regulatory requirements? I have made my slide deck very short in brief, which will contain the basic requirement which will student will able to understand basically. It is not in detail, so it will not make you boredom also, because I understand this is a post lunch session and sometimes we may get lethargic after hearing so many sessions. So it will be short and sweet. And in continuation to these biological product regulatory requirements, I will be going through a walkthrough for the regulatory. So what are the basic requirements in the regulatory for any kind of product approval? So that will be my continued lecture after this. So good afternoon all. Hope just before starting with the presentation, just want to confirm whether the slide deck is visible and I am audible properly. Can you please confirm, sir or madam?
Yes, madam.
Okay. And which language is preferable?
Both English and Marathi language, madam.
Okay, sir. Thank you. Okay. So let's have a look into regulatory requirements for biologics product approval. So as you all must be knowing, biological product we are using and available in the market. So in layman's language, biological products are, for example, insulin. It's a biological product. Any kind of sterile anti-cancer injection is a biological product. Monoclonal antibodies, vaccines, these are biological products. So the topic will cover what is the biological product, that is introduction, source and type of biologics, that is biological product, what will be the difference between biologics and chemical drug, what are the regulatory authorities for biologics, how there is a development and approval process for this biological product, what is biological license application, that is generally called as BLA in terms of regulatory submission, comparison between US and Europe guidance for biologics, and ultimately the conclusion session. You can enter your queries in the chat box. If relevant, I can reply to you. It's possible online, or I can share the reply to madam as well later on. Shall I continue? Okay. So let's get introduced. I think you all are aware about what is a biologics. Still, I just want to brief you what is biologic or biological products. These are the products which are manufactured, extracted from, or semi-synthesized from biological sources which are regulated by FDA. So, making the product usable to our customer or patient, those products should be regulated or controlled by any regulatory authorities, that is our FDA. These are used to prevent and cure disease and medical conditions, especially cancers, diabetes, etc. And our vaccines, which are immunological products. These are large, complex molecules produced through biotechnology in living system, such as plant cell, microorganism, animal cells, etc. These are heat sensitive, heat labile. These are susceptible to microbial contamination, and hence it require aseptic manufacturing principle from initial process. So in brief, if we understand, the biologics are sterile products and it requires aseptic manufacturing. Certain examples I have mentioned over here, like Botox vaccine, pegfilgristins. These are some example of marketed biological product. If you wish to ask any question, you can ask in this forum also. So let's see what are the source and types of these biological products. So as you understand, there are various sources of biological products and the types of that. So any kind of source which is originated from biological, like mammalian cell culture. So blood derivatives will come under mammalian cell culture. Bacteria from which produce vaccine, then insect cell culture, this is for the allergic products or allergenic extract preparation. Then plant cell culture, which is for the whole blood. Then yeast as a fermenter for the protein. Then avian cell culture for cellular and gene therapies type, and human cell for xenotransplantation products. So these are certain sources which are being used in manufacturing of biologics, these types of uh, products. So let's see what is biologics versus chemical drugs. So you must be thinking how the biological drug is different from chemical drugs. So chemical drugs are allopathy and all those come under chemical drugs. So let's go property by property. So if we consider the size of biological molecule, it is large and its structure is complex, opposite to chemical, which is small and simple. So, if you understand, the complexity and the manufacturing processes required for the biologics are very tedious. As being experienced in the biologics, I understand that there require n number of activities during research and development for the biological product. There are chances of failures, so many failures, and of course, it is a very tedious. and also challenging to produce or make the product available for the production after research and development. Also, it requires preclinical, clinical trials, same as chemical drugs, but these are very complex because it includes the sterile manufacturing. Considering the stability property, these products are unstable and hence specific stability condition is required to maintain this product. Comparatively, chemical drugs, those are stable. The modification in the cell or tissues we can do, or genomics modification. There are multiple options in the biologics, and in chemical drugs these are well defined, so it is a specific. For the manufacturing, it is difficult to ensure identical copy. Suppose I'm produce insulin right now, but I'm not sure whether I can produce the same kind of insulin in the uh, research and development activity. However, in chemical products, the identical copy can be made. So these are generic products, we say. Characterization. In case of characterization, it is difficult to characterize because these are large and complex molecules, and chemical drugs are very easy to characterize. So the instrument used for the characterization for biological products are also very sophisticated. So what are the regulatory authorities for these biological products? So enter for biological evaluation and research, that is CBER. Then FDA regulatory authorities for product approval, mainly US FDA and Europe, that is EMA, are mainly stringent authorities which uh, emphasize on the strict regulation for marketing authorization of these biologics. And of course, UK and MHRA. So let's go through the development and approval process. So if I want to develop any biological product, how I can go ahead and how I can go for the approval process. So we have to consider the advertising and labeling, then investigational new product drug application we have to pass, then expanded access, pre-market approval is required, then biological license application is required to FDA, then new drug application process is started, and the biological approval by the year of that particular submission year or submission period for that biological product it happens. So this is just a brief I have explained you. So the main role is played by this BLA, that is biological license application, which is a mandatory process for getting the approval for the biological product. It is a request for permission to introduce biological product into market. It is regulated by 21 CFR Part 600 and 800. It is generally applied for vaccines, allergenic drug product, cellular and genetic therapies. And to apply this BLA, the form 365H is to be applied, or the application through form 365H is to be done. And the requirements of these forms are, we have to put the applicant name. For, let's take an example of any biological company. So we have to put the name of that company, then which product is being manufactured and its manufacturing information, its preclinical studies data, and its labeling information. So especially in the labeling, it comes the storage conditions, shelf life, those are the things, and how to administer. So those things are coming to picture. So these are the basic requirement which are to be put front while applying to 365H form. So if you see the guidelines requirement, that is comparison of guidelines of US FDA and EMA, that is European Marketing Authorization. So what are the requirements? Let's go one by one. So if we consider regulatory body US FDA, it has adopted S6 guidance, and EMA has adopted ICH S6 guidance. For approval standard, it is they require application through BLA process, that is biological license authorization, and for Europe it is required MAA, that is marketing authorization application. So it is similar to our other drug or formulation, that is a chemical drug, also the same kind thing. Only thing, the regulatory body S6 guidance is different for biologics. For these, preclinical studies are required, especially the species are different, mentioned in the USFDA, which has chimpanzee study, and in EMA, the requirement is for rodent and non-rodent study. Then clinical studies, only pharmacodynamic is being performed as per USFDA requirement, and for EMA, pharmacokinetic as well as pharmacodynamic. For clinical studies, it involves three phases, all three phases in USFDA requirement, and for EMA, it must comply as per GCP E6 guideline. So these guidelines are easily available on any browser, in the Google Chrome or any other browser. You can search and navigate to this guidance. If you wish, you can write it down right now only, or you can type it somewhere. So for you also, it is easy to navigate these guidances for your better understanding. For clinical studies, there are total phase 3 trials they perform, like phase 1, phase 2, and phase 3. So if we consider the US FDA requirement, so phase 1, it involves patients for maximum tolerated dose and not the normal human volunteer. Then it studies for product metabolism, pharmacology, and safety at high dose. However, as per the EMA requirement, it studies initial safety, tolerability, pharmacokinetic, and pharmacodynamic activity. For phase 2, in US FDA, it requires safety evaluation and short-term adverse event study. And for EMA, it requires safety and efficacy study. For phase 3, it begins if preliminary evidence of effectiveness is shown in phase 2. That means if there is any effectiveness is shown in previous phase 2 trial, then it goes with the phase 3 trial. And in EMA, there is a therapeutic confirmation and dose response relationship. So the US FDA is more stringent, I can say, comparatively to EMA. However, EMA performs the PK and PD activity as well, and safety and efficacy study over here. So this is a overall comparison with respect to US FDA and EMA guideline for the biological product manufacturing. Let's move to the next. So this is overall, I have given you the brief idea how we can, what is a biological product, how it is being marketed, and what are the requirements of the regulatory to market the biological product. So let's have a conclusion of this topic, and then we can move to the next topic. So to conclude this, the drug approval in US and Europe are most demanding in the world nowadays for this biological product, and the main objective is to safeguard the public health. Of course, these are the sterile product, and those are getting directly into administered into the bloodstream. Systemic administration is there. Okay, so the public health, to safeguard the public health is the main objective of these regulations, and the regulatory authorities to ensure that biologics manufacturer must comply their regulations. So this is a main motto of these regulatory agencies for biological product approval. So until and unless we do not go, or we do not submit these requirements to the regulatory, we don't get approval for our product. It's very stringent to get the approval of the biological product in a US and Europe market especially, and challenging as well. So this way I have concluded with the biologics, and I wish to move to ahead with other topic, that is overall regulatory requirement in pharmaceutical manufacturing. and this was specific to the biologics. So meanwhile, if you have any question, you can post it. Okay. Okay, so let me start with regulatory affairs walkthrough. So what is regulatory affairs? Regulatory affairs is a comparatively new profession. So of course now it is almost well versed profession which is developed from the desire of governments, that is these are the regulatory bodies, to protect public health by controlling the safety and efficacy of products in areas including pharmaceutical, veterinary medicine, medical devices, pesticides, agrochemicals, cosmetics, complementary medicines, etc. Of course, biological also come as a part of pharmaceutical. Regulatory affairs in pharma industry, it may be defined as the interface between pharma company and the regulatory agencies across the world. So as you might have understood from my previous presentation that there are certain requirements from regulatory bodies that those expectations they put forward for pharma manufacturers and that we have to fulfill to sell or market our product in that particular regulated countries. How the regulatory affair got evolved? There is one statement that all substances are poisons. However, there is one which is not a poison. The right dose differentiates a poison and a remedy, that is a medicine. So whatever we take it in the proportionate quantity, that is our remedy. And if we go beyond that or out of that box, then it can be harmful to anything. So it's in general, it's a it's a natural thing that anything we do which is beyond limitation or beyond capability, sometimes these are sometimes tedious or harmful also, and the same principle is applied to the medicine as well. And why to regulate? Why to regulate these kind of products? Like to ensure quality, safety, and efficacy of drug product in order to assure the continued protection of public health. So no product is completely safe in all circumstances, but there is a moral and legal requirement which are required to be fulfilled to make the product very much safe and efficacy to be maintained in the patient. So why does a DRA, that is Drug Regulatory Affair Department, is required in pharmaceutical organization? So you might be thinking, I will join a regulatory affair after passing out my B Pharm, M Pharm, or passing, completing my doctorate and all. So that is a good vision. If you are wishing or willing to go that, because there are multiple regulatory bodies and their requirements are getting stringent day by day, and for which the resources are required in the pharmaceutical companies, in especially in the regulatory affair department. So these are the legends or key department. This is a key department between pharmaceutical manufacturer and in the regulatory body. So let's take an example of US countries. US countries put their requirements to a particularly company. Let's take example of Cipla for their respiratory product, and if they fulfill the requirement of the US regulations, they can market their product in their country, and the sale will be more in their country. Similarly, other countries' regulations to be followed, and those are to be fulfilled by the manufacturer, and their product will get approval, and that will get marketed and sell out in that particular country, and there will be a business to our pharmaceutical manufacturer. So let's have a walkthrough why it is required, why the regulatory affair department is required. So pharmaceutical industry is most regulated of all the industries, as you are aware, because we deal with the patient safety and product quality. Regulations are put in order to develop the most efficient and safe pharmaceutical product. It takes more than 8 to 15 years to develop a new drug product, and it costs more than $800 million. So you can imagine how much it is getting cost. And considering the financial attrition or financial market increase growth every year by year, so this expense is more and more, and also the tenure required to develop is also more. So regulatory affairs, it provides insight or a guidance into this development through agency wisdom collected in guidance or previous experiences, market precedences, and it hence helps to reduce a number of development failures. So, what is the main role of the regulatory affairs? They gather a data, they make a survey of the previous any kind of marketed product or any kind of derivative or being done before, or any kind of survey done, or any case studies are there. Based on that, they start searching and they provide the information to manufacturer. Based on that, manufacturer evaluates the requirement of that particular country or region, and then we start manufacturing the required pharmaceutical product for the particular country. So what is a product life cycle? So for getting the things to be done as a part of regulatory, we should know what is a product life cycle. So from the beginning of the product till its marketing in that particular country, and it is a regulatory affair perspective. So there are three phases, that is development phase, then it's approval phase and post approval phase. So in development phase, as I just now spoke to you, like there is a scientific advice required, then clinical trials required, then project management is required to make these trials on time or before time, then the product information is required. So gathering all these things, the product is being developed. Let's take simple example of acetaminophen tablet. If I want to market my tablet in any kind of regulatory, like I want to market in Canadian countries, those as an antiviral or antipyretic product. So I have to gather and I want to make in some other um, novel drug delivery system. So I have to gather these requirements, the product information, other dosage form, how our product will be more helpful, more suitable, and more controlled for the disease. And that information we have to gather in the development phase. We have to work with the preclinical as well as clinical trials. And then once all the data will be gathered, it will go for the approval. So approval is nothing but approval in that particular regulatory body. So if I want to submit my approval procedure documents for the approval to particular country's regulation, I have to apply through the proper procedure, application procedure. I have to have a authority meetings, and then I have to go with e-submissions. So in ancient days, ancient means in the past days, there are the submissions, manual submissions through courier and posting and etc. However, now in the industry, we there is a gateway for e-submissions. So we submit dossiers or the required marketing requirement to the electronic gateway to regulatory bodies or regulatory authorities to particular country. So it will be more easier, more faster, and less time consuming to get the approval for that particular product. And in case if it is a first to filing, so if we have a electronic submission gateway, it will be more easier, convenient to submit our product details to that particular regulatory. So what comes after approval phase is, once you will get the product approval, there will be a post approval phase. So that is a life cycle management phase of that product, correct? So in the life cycle management phase, there are certain things like market complaints come, sometimes product recall come, any observation or regulatory queries come from the particular region, then that is to be handled in the post approval phase by regulatory affair department, which is called as post approval commitment. So these are the three phases of life cycle. Let's see which departments are assisted by drug regulatory affair department. So internal departments of particular XYZ pharma company are, which are connected with this drug regulatory affair department, are product development, clinical research and trials, licensing, manufacturing, quality assurance and control, and marketing. And nowadays business development also has been started along with product development, so they see how to expand a company's business in particular country or in with the particular regulations. So the scope to our new candidates is more in pharma company. So the basic requirement for the regulatory is to know what is dossier or DMF, which is a heart of our submission of any product approval, for any product approval. So let's go see what is a dossier. So dossier is nothing but a collection of or a file of documents that contain all the technical data of pharmaceutical product to be approved or registered or marketed in a country. It is most commonly called as registration dossier. So in US countries, it is called as new drug application, that is NDA, and in Europe countries, it is called as marketing authorization application, that is MAA. So generally, when you will join a regulatory department as a fresher, you will come to know these words on your ear, like what NDA, MMA. If you are aware about these words, then it will be easy for you to understand these terminologies. So DMF. So there are two things. The dossier is for the finished product and DMF is for the active ingredient, active pharmaceutical product, API. So DMF is nothing but drug master file. There are multiple types of the DMF. So type 1 is for the manufacturing site facilities, operating procedure and personnel, which is no longer now applicable. Then type 2 is for the drug substance, intermediate and material used in their preparation or drug product. Type 3 DMF is for packaging material. Type 4 is for excipient, color and flavor or material used in their preparation. Type 5 is for FDA accepted reference information. For United States Drug Master File, it is called as US DMF, and for Europe, it is European Drug Master File, that is EDMF or ASMF, that is Active Substance Master File. So there are two key things you should remember. That is, for the finished product approval, we require a dossier to be submitted, and for active substance, we require DMF to be submitted. Suppose, let's take an example of anti-diabetic drug metformin. So I want to make a sale of metformin in Europe country. Then I will go ahead by filing a ASMA for EDMA for that, for the metformin. And if I want to market metformin sustained release tablet in Europe country, then I will go with the MAA, that is Marketing Authorization Approval Application for Europe country. This is the difference between dossier and DMF. So now you will hear this terminology, that is what is CTD or ECTD. So as I explained, that is a electronic submission gateway which is being used in regulatory for submission of this approval, for the approval, dossier and DMA. So let's check, let's go with the CTD. So what is CTD? Is a common technical document. It's nothing but a file of all information of the products history, geography, entire thing. Everything is there in the CTD. It is in formats which is set by ICH guideline, which was agreed by regulatory agencies of Europe, Japan, and US. Its electronic version is called as Electronic Common Technical Document, that is ECTD, which is nothing but Electronic Submission Gateway. So now you must be thinking in your mind, so how it is linked with dossier, what is dossier, DMF, what is ECTD or CTD. So we have a package ready with dossier or DMA. We have a electronic submission gateway, that is ECTD. There are certain softwares in market available which the pharmaceutical industries are being used. Those are being used in the pharma industries for the e-submission. For example, Pharma Ready is one of the kind of this software for e-submission. So we have to collect all the data, we have to scrutinize all the data, we have to check with all cross-functional departments of the company, and then we have to submit to the regulatory body through this e-submission gateway. So the submission is more faster, feasible, and easier. So what are the benefits of the e-CTD submission? So most relevant is it can reduce the time for approval. So how it will reduce the time for approval? Let's see. Because we are submitting it through the e-submission gateway, so it is a immediate submission. If you sit at home, that time also you can submit it if you are having that ECTD gateway of that particular regulatory site. So our submission process is very fast. Then improved handling and archiving of submission, that is both sponsor and FDA. So that means if sponsor and FDA, regulatory body, for them the handling of the documents, it's very easy. And what is archiving is nothing but the data is residing in that particular gateway, that is software or application, and where the handling of any kind of document or information is easy anytime to that regulatory body. The search functionality and increased tracking availability. So there is a fixed dossier modules in that the particular data goes and reside, and hence the search functionality such as hyperlinking is there, bookmarking is there. It will make the regulatory people more easy to identify any kind of document before its product approval. It allows for repurposing of documents for submission in other regions. So this is the most beneficial, or it is called as a leverage, that in other countries apart from US or Europe or Japan or MHRA, other countries also there. So there also you can submit these ready-made dossier to other countries. Accessibility to documents across modules. So all the documents are accessible across all the modules. So what are modules we will see in the next slides. Then submission via ESJ allows immediate receipt by FDA. So what is ESJ is Electronic Submission Gateway, and it allows you immediate receipt by FDA, and it improves reviewer efficiency because everything is in the table of content. Reviewer can easily pick any document he or she wants to review for the product approval, and based on that we get a product. It reduces the approval time for the product. So these are overall benefits of the ECTD. Initially, what company used to do, they used to file these modules in on paper, and those files are being couriered or posted through regulatory agencies. To reach those documents to the regulatory agencies, it used to take a lot of time, of course, and hence the product approval, it was very time consuming. So let's see the structure of ECTD or CTD. It's a common technical document structure, which is a module. There are 5 modules: Module 1, Module 2, Module 3, Module 4, and Module 5. Now you are looking this pyramid on paper, And the same paper is presented now in application or software with hyperlinks, bookmarks. So for the user or the customer, it is very easy to submit those documents through these modules. And of course, there is a validation rule. If we attach any incorrect document, then the software throws the validation error also, and hence it is 100% quality controlled submission gateway, and hence it is very approved submission process in the regulatory market. So what is the module 1 content? So it contains administrative and prescribing information, which is a basic information on that particular drug product or APIs. Then module 2, it is a summary module, which is a quality overall summary. These summary are for non-clinical overview and clinical overview, and non-clinical summary and clinical summary. Module 3 is for quality data. Quality data means quality laboratory, quality control testing, all those data. Module 4 is for non-clinical study report, and module 5 is for clinical study report. That is pharmacokinetics, dynamics, then bioequivalence in case of generic drugs approval. So those are the fixed 5 modules for the ECTD or CTD, we can say. Let's go each module one by one in short. So module 1 is administrative information module, and it is region specific. Region specific means what? For US, its requirements are different. For Europe, its requirements are different. For Canada, its requirements are different. For Japan, its requirements are different. Means how we are presenting the product information as an administrative information as per the regional requirement, and it should contain the documents which are specific to that each region. So I have jotted down here for US and Europe. So for US, that application form 356H is required. Then its label, proposed label, what the manufacturer is proposing. Then its patent certification or information if it is to be patented. Then it is department certificate or any letter of authorization, or its DMF letter. Then its labeling text. These are the basic requirement. If we correlate these requirements with biological, these are near about same for the BLA, that is biological license application. For Europe, it is an application form, summary of product characteristics, and labeling text and mockups. These are the basic requirement for the module 1. For module 2, as I explained you, it is a summary module. So it contains overall 7 sections in following order. Like it's a CTD document module, that is module 2 to 5 summary is there. Then common document introduction is there. Quality overall summary is there. Non-clinical overview, clinical overview, and non-clinical summary and clinical summary. So if you see any of the regulatory submission on Google also, you can find these wordings or this terminology, and you can go through it in detail if you want to understand further. Module 3. So module 3 is a quality module. It contains the table of content for module 3 and body of data. So what is body of data? That is the details of the drug substance which is used to formulate a drug product, then details of drug product, then any kind of appendices, regional information, and any literature references. So regional information means any kind of case studies performed with this drug, any kind of disease condition which are cured before previously in this drug. All these things are the regional information to be presented along the in this module. Module 4, it is a non-clinical study report. It also contains the table of content on module 4, the study report of pharmacology, pharmacokinetic, and toxicology along with literature references. So this is a part of non-clinical. So what is module 5? So it is a clinical study report with TOC of module 5. TOC is nothing but table of content. So when you see any dossier, there is a TOC, table of content, for each module, and from that it is easy to understand which document has been submitted in that particular dossier. Then the tabular listing of clinical studies, then clinical study report for BAB studies, that is biopharmaceutical study, report of pharmacokinetic study, report of pharmacodynamic study, then report of efficacy and safety study, report of post-marketing experience, and any case report forms and individual patient listing and literature references. So based on the past year experiences, this data has been captured and to be put forward in the module 5 and conducted clinical studies. These are overall understanding of module 1 to module 5 of any kind of dossier. So what is ECTD? We have briefly seen in previous slide what is ECTD. Let's study in detail. Like ECTD is an electronic version of common technical document, and it's called as electronic common technical document. It is composed of two types of specification, that it's content specification, which is defined by ICH guideline, and technical specification, which is defined by electronic software's common technical document. So content specification is nothing but the administrative specification, and technical specification is from module 2 to 5. So ECTD is highly recommended by US FDA for new drug application, biological license application, DMF application, and investigational new drug filing. So usually US countries prefer this submission gateway or faster filing of any kind of product in their country. So what is ECTD characteristic structure? So all modules 1 to 5 have granulatory option. PDF document is landscape with the bookmarking, hyperlinking, and present of all documents. And in post compilation, we have to see validate the compiled ECTD, and we have to check the broken links as well. And after compiling everything, we have to submit it to regulatory bodies through e-submission gateway. So these I have explained you about the CTD and ECTD. Now let's have a look what is the role and scope of regulatory affair profession. So hopefully you will have you have have this certain understanding of the regulatory bodies as well as regulatory departments in pharma industry. So let's understand now what is will be the profile of the regulatory affair uh, person in any kind of pharma company. So to keep a track of ever-changing legislations, so what are the legislations means the regulatory requirements of any country or any regulatory body. So we have to keep on the changing or updating or upgradation of the regulatory requirements for the product submission. Let's take an example. I want to file my biological product in US market, and there are any enhanced requirement from the US, then we have to get the updated one. We have to keep a track for those legislation, and then we have to keep us updated according to their legislation. Then registration documents to regulatory agencies, that means through this submission gateway, to give strategic and technical advice to R&D, product, QC department, etc. That is cross-functional. So how it the strategic and technical advice? Let's take example of the QC department. So QC department does the analysis, wet chemistry, dry chemistry, etc., etc. So while submitting, regulatory may ask why there is a OS or OOT observed, right? So then again we have to go with the production. So the everything should be justifiable, and then only we can give uh, the answer to regulatory. That means the regulatory affair person is a liaison between their pharma company and to the regulatory bodies. Okay, and hence it acts as a liaison with regulatory agencies. So these are the basic profiles with the RA role. What is the scope? of this regulatory affair. Of course, there is a vast scope in any kind of pharma company, though it is a biologics, cosmetic, veterinary product, everything. So there is a vast scope to the regulatory affair profile that the employee will get a job in industry or government regulatory agencies such as DCGI, CDSO, or if you wish to go out of India, then it will be US FDA is there, UK MHRA, WHO. In academics for pharmaceutical, you will have a lecturership in the academics as well. With medical device, it plays as a consultant, regulatory consultant. Then nutritional product, biologics and biotechnology, and in vitro diagnostic. And of course, the knowledge is a key role over here. That is a good knowledge of all documents related to respective countries' guidelines and its update. So this is a basic requirement to any kind of regulatory affair professional. Okay. So let's see what are the regulatory agencies. We were speaking so many times that about the US and Europe only. So let's see who are the other regulatory agencies apart from US and Europe. So let's start with our country, India, that is CDSO. And this is its website for CDSO. Then USA, that is usfda.government. European, it is EMA. Then for UK, it is MHRA. Then Australia, it is a TGA government. Then for ICH, then Canada, South Africa, Singapore, and Brazil. Those are the websites I have mentioned over here. And you can easily find those websites on Google, and the requirements are mentioned over there for the any submission of BLA or MAA or any new drug submission. So let's see region-wise, country-wise regulatory bodies in the world. So if we want to file our submission for US countries, so countries USA, where it is called as FDA, that is Food and Drug Administration. For UK countries, it is MHRA, that is Medicines and Healthcare Products Regulatory Agency. For Australia, it is TGA, that is Therapeutic Goods and Administration. For India, it is CDSO, that is Indian Central Drug Standard Control Organization. For Canada, it is Canada Health Canada. For Europe, it is European Medicines Agency. For Japan, it's Ministry of Health, Labour and Welfare. And for South Africa, it is MCC, that is Medicine Control Council. So whenever you will appear for any fresher interview for regulatory, you should know the full forms of this regulatory body along against the specific regulated countries. All these things you should know basic information about CTD, that is common technical document. You should know basic information about ECTD, which are the commonly software being used in the currently in the pharma industry. Those information you should know. Then what are the modules basically that details you should know, that is one module 1 to 5, and what each module has a content, that also you should know while going for the interview in any regulatory affair department. Okay. And why the regulatory affair department is important, that also you should know. So let's come to the conclusion that in today's competitive environment, time is required for medicinal product to reach market approval is critical. That means we have seen it takes around 8 to 15 years to get to develop one product, which is which leads to company's success and growth ultimately, right? And also the implementation of guidelines and legislation. So legislation keeps on changing, will improve companies' financial growth and ultimately patient safety. So when we file our product, so if there is a first to file of your product in US country, then you will get faster approval and your business will start, your product will get marketed, it will get sold out in US countries, and your business for your manufacturer will be more and more. And of course there will be a patient safety to that particular region. And also it is important to keep the product in market for long time. So once we capture that particular regulated market, it will, our product will keep in that market for long time, 3 years, 5 years, 10 years. If it is a patented drug, it's more years, and it ultimately it is a beneficial to pharmaceutical manufacturer. So this is overall a walkthrough of the regulatory affair, and hope you have understood this topic. Uh, so from my side, I have done.
Thank you, ma'am. So, may I can conclude our session. Before that, I would like to talk about the for participant to ask the questions.
Sure. It is better if you can pass the question to Dr. Chikle sir, so that he can convey it to me. Because
Ask your question.
Why some of the medicines are banned in other nations but still are used in India?
your voice is clearly audible, sir.
Okay.
There is a echo.
Hello, sir. Hello, sir. Am I audible?
Yeah, you are.
Sir,
Yes, ma'am.
Why some
madam,
of the
एक क्वेश्चन
medicines
है.
are banned in all nations but still are using in India?
Sorry. Can you please come again, sir?
Ma'am, can you please quote about reimbursement from consumer point of view?
Okay. So reimbursement from consumer
Hello,
point
madam.
of view.
Hello,
हाँ,
madam.
बोला ना, बोला. What is your question, madam? Can you please come again? And may I know your name, please? And you are in which standard? I mean to say, which year of pharmacy? Hello.
Am I audible, ma'am?
Yes, sir.
I'm a student, so I have one question.
Hmm.
Why
You
some
are
of the medicines are banned?
sorry, you are in which year of pharmacy?
UG second year, micro BSc.
Okay, you are in micro BSc. Okay, fine. Okay. Yeah, tell me.
क्वेश्चन विचार म्हणली का एकदा अजून.
हाँ, बोला. प्रश्न बोला तुमचा. सांगा. बोला.
Why some of the medicines are banned in other nations but still are using in India?
सर जरा मोठ्याने बोलाल का? आवाज येत नाही काहीच.
30 सेकंद देता.
Why some of the medicines are banned in other nations but still are using in India?
Why some of
Ma'am,
the
can you please quote about reimbursement from consumer point of view?
reimbursement
Reimbursement.
means what? म्हणजे काय? हॅलो. तुम्हाला असं म्हणायचं आहे का की कन्झ्युमरला काही इश्यूज असतील तर काही रिएम्बर्समेंट कशी असणार असं प्रोडक्ट रिलेटेड?
हॅलो
हा
मॅम, रिइम्बर्समेंट म्हणजे परतावा. जर एखादा ड्रग जर आपल्याला इफेक्टिव्ह नसेल तर
रिएम्बर्समेंट ना, रिएम्बर्समेंट म्हणताय ना तुम्ही, परतफेड बरोबर, ग्राहकाच्या दृष्टिकोनातून परतफेड बरोबर ना. हा, या केसमध्ये असं असतं की प्रोडक्ट स्टॉप केल्या जातात किंवा त्याचे रिकॉल केलं जातं. आधी कस्टमर किंवा मार्केट कंप्लेंट्स आयडेंटिफाय केल्या जातात. सपोज तुम्ही एखाद्या प्रोडक्ट, सपोज
येस मॅम.
ऑइंटमेंट आहे,
येस
करेक्ट.
येस.
त्या ऑइंटमेंट जर तुम्हाला त्याच्यात काही पार्टिकल्स आढळले तर तुम्ही त्या पर्टिक्युलर XYZ कंपनीला कंप्लेंट करू शकता. कन्झ्युमरचा जो फोरम असतो त्याच्यातून तुम्ही कंप्लेंट त्यांना द्यायची असते आणि त्यांची ती स्टँडर्ड ऑपरेटिंग प्रोसिजर असते, तर प्रत्येक कंपनीची असते. सपोज तुम्हाला ते पार्टिकल्स आढळले आणि तुम्ही जेव्हा ते यूज करत होता, तुम्ही ते परचेस केलं मेडिकल स्टोअरमधून आणि तुम्हाला तसं आढळलं, तर यू कॅन राईट अ कंप्लेंट to the through the complaint module to the website to that particular company. Company will take that on priority considering its seriousness and criticality. आणि त्याचा ऍडव्हर्स इव्हेंट काय असू शकतो त्या बेसिसवर कंपनी तुम्हाला काही तासांमध्ये किंवा काही दिवसामध्ये तसा रिस्पॉन्ड करतो आणि मग ते प्रोडक्ट किंवा ती बॅच असते किंवा तो लॉट असतो त्या प्रोडक्टचा, तो मार्केटमधनं काढला जातो. त्याचबरोबर त्याचं आधीच्या बॅचेसचं इन्व्हेस्टिगेशनही केलं जातं आणि जिथे जिथे त्या बॅचेस गेल्या असतील त्या सगळ्या होल्डवर गेल्या जातात. त्याला प्रोडक्ट रिकॉल असं म्हणतात. आणि त्या बेसिसमध्ये त्या रिकॉल, कधी कधी प्रोडक्ट रिकॉलही होतो त्या होल्डवर गेल्या असतील आणि काही मेजर त्याच्यात इम्प्लिकेशन्स असतील, देन द प्रोडक्ट गेट्स रिकॉल्ड, असं असतं. बरोबर, ओके. नेक्स्ट क्वेश्चन. अजून काही
Yes, ma'am.
क्वेश्चन आहे का?
Why some of the medicines are banned in other nation but still are using in India?
Good question. So in this case, Indians are more powerful, I can
Thank you
say,
ma'am.
or yeah, welcome. So they are more stringent to the response to any kind of medicines. So as you see, how we live in our country, it's a versatile country. Our nature is very versatile. There are multiple seasons. There is a seasonal variation is more and more, and then climatic zones are also different. Our food habits are also different. So in such cases, we are not more prevalent to the responses of any kind of drug, such certain drugs like NSAIDs or the nimesulide. Let's take an example of nimesulide. So we are not prevalent and we are responding very well to those drug treatments in India. And in other countries, considering their lifestyle, genus and species, their responses to drug, their adverse events are more and comparative to India, and hence those drugs are banned in other countries.
Okay, ma'am.
So that's why the studies are important. That is kinetic studies, dynamic studies, then clinical studies. So the clinical trials are very important, and those are conducted in all regions of the world where we want to export our product from India, right? So in India also we conduct these things trials, and based on that data, we identify whether the drug is suitable to which species, which gene, or which region, ideally. Okay. Yes,
Okay,
please.
ma'am. What is the meaning of pharmacokinetics and thermodynamics?
Yeah. So pharmacokinetic is nothing but the what drugs do on our body, and pharmacodynamic is what is body does on our drug. In short, we can say.
Okay, thank you, ma'am. Ma'am, another one question is, what is time span for production of vaccine?
Of course, as I said you, it takes around 8 to 15 years. Production of vaccine, you can say once the recipe is finalized, after the development. For development, it takes around 8 to 10, or it can be 15 years also, because it's a sterile product, and it's a complex molecule. And for the production, it takes around a month, I can say, because month or 2 to 3 months, because there are the fermentation cycles, upstream and downstream processes to be done. Then it's IPQC to be performed, it's IPQA to be performed, and then it's quality control, and then it's packing, sterile packing, and then it's release. So it takes around 2 to 3 months, we can say, for the production.
Okay, ma'am. Thanks.
So any other question, or can we wind up?
Any question? Participant can ask the question.
हॅलो सर.
Yes.
सर मी मायक्रोचा स्टुडंट आहे, मग मला रेग्युलेटरी अफेअर्सशी रिलेटेड कुठला कोर्स करता येऊ शकतो का एक्सटर्नली असं?
हो, का नाही करता येणार? रेग्युलेटरीचे बरेचसे कोर्सेस आहेत. असं काही नाही की मायक्रोचे स्ट्रीमचे स्टुडंट्स रेग्युलेटरीमध्ये नाही जाऊ शकत. उलट मायक्रो इज अ व्हेरी क्रुशियल टॉपिक इन एनी काइंड ऑफ मॅन्युफॅक्चरिंग. तर तुम्हाला जर त्याचं रेग्युलेटरीचं नॉलेज असेल विथ रिस्पेक्ट टू मायक्रो, तर इट इज ऑलवेज हेल्पफुल इन रेग्युलेटरी ऑल्सो. तुम्ही कोर्स कोणतेही करू शकता रेग्युलेटरीचे.
ओके, थँक्यू मॅम.
आणि जर तुम्हाला कोणतेही कोर्स करायचे असतील, तर यू कॅन रीच आउट टू मी थ्रू युअर ऑथॉरिटीज. दॅट विल बी हेल्पफुल टू यू.
मॅम, US मध्ये PhD वगैरे करण्यासाठी रिक्वायरमेंट काय आहे?
US मध्ये PhD करण्यासाठी तुम्हाला आधी इकडे B.Pharm कम्प्लीट झाल्यावर जर तुम्ही MS करायलाच US मध्ये गेलात, तर तुम्ही तिथे PhD करायला इझी पडेल. पण जर इथे M.Pharm करून मग तिथे PhD करण्यासाठी गेलात, तर तिकडच्या स्टेट लेवलच्या एक्झाम्स असतात, त्यांच्या एंट्रन्स एक्झाम असतात. तर जसं की तुम्हाला ऑफकोर्स GRE आणि TOEFL तर द्यावी लागतेच. US मध्ये किंवा कुठल्याही आउटसाइड कंट्रीजमध्ये फर्दर स्टडीज करायचे असतील, तर तुम्हाला GRE ग्रॅज्युएट रेकॉर्ड एक्झामिनेशन म्हणून ही जी एक ॲप्टिट्यूड टेस्ट असते, त्याचं प्रिपरेशन करावं लागतं काही महिने. आणि त्याचबरोबर TOEFL ही लँग्वेज स्किल रिलेटेड एक तुमची एक्झाम असते. या दोन्ही एक्झाम तुम्हाला द्याव्या लागतात. त्याच्यात तुमचा ठराविक स्कोअर आला की तुम्हाला जिथे ज्या कोटाप्रमाणे ॲडमिशन मिळतं, जसं की आपल्या इथे काही कोटाचे लिमिट्स ठरलेले असतात की एवढे पर्सेंटेज मिळाले की इथे ह्या कॉलेजला किंवा या युनिव्हर्सिटीत मिळणार. तसंही US मधल्याही बऱ्याचशा युनिव्हर्सिटीज आहेत, तिथे तुम्हाला ॲडमिशन तुम्ही घेऊ शकता. तसंच त्याबरोबर तुम्हाला तुमच्या फायनान्शियल रेडीनेसचाही विचार करावा लागेल, कन्सिडरिंग युअर फायनान्शियल बॅकग्राऊंड, तुमच्या फॅमिलीचं फायनान्शियल बॅकग्राऊंड. बिकॉज आपल्याला तिथे काही ॲज अ सेक्युरिटी डिपॉझिट म्हणून तिथे काही अमाऊंट तिथे दाखवावी लागते बिफोर टेकिंग ॲडमिशन इन एनी युनिव्हर्सिटी. हा सगळा ओव्हरऑल विचार करून तुम्ही तिथे ॲडमिशन घेऊ शकता. आणि तुमचा स्कोअर जर चांगला आला असेल या 2 एक्झाममध्ये, तर तुम्हाला इझिली चांगल्या ह्याच्यात ॲडमिशन मिळू शकतं. आणि त्यांचा गायडन्स करणारेही आपल्याकडे बरेचसे कोचेस आहेत, जे फॉरेन स्टडीजसाठी गाईड करतात. तुम्हालाही माहीत असेल.
ओके मॅम, थँक्स.
अजून काही प्रश्न आहेत का कुणाला? So can we wind up the session now, sir? Chikre sir?
I hope there is no more questions. Last session of this workshop. Good afternoon, everyone. On behalf of Venkatesh Mahajan Senior College, Dharashiv, I extend my heartfelt gratitude to Pennegar Madam for her insightful and informative session on regulatory requirements of biological product production. Madam, we truly appreciate the time and effort you took to share your knowledge with us today. Your expertise and experience in this field are invaluable, and we are grateful for the opportunity to learn from you. I would also like to extend my thanks to organizer and of course a big thank you to our audience for your participation and engagement. Once again, thank you, ma'am, for an enriching sessions. Madam, we look forward to more such interaction in the future. With kind permission of the organizer, this session will here conclude for our side. Thank you, madam.
Thank you, and I also want to appreciate your patience, your senior authority, and all staff and your students' patience for listening me for last 1.5 year, I can say. And I will, of course, I'm happy to give you more and more sessions online as well as offline. I hope I will meet you in next session. Online over the offline at your college premise, and I will be really happy to meet you, your authorities also, and everyone at your place and your college and university. So I'm thanking to Venkatesh Mahajan College for giving this opportunity to present this topic today. Thank you.
Thank you. Thank you, ma'am. Thank you.
Hello. Hello.
हाँ सर. हाँ सर मॅडम.
आवाज येतो का?
हा येतोय आवाज. मॅडम होतील जॉईन 5 मिनिटाचं.
ओके मॅम.
5 मिनिटात.
येस. हो हो मी
टेक्निकल.
मी ऑनलाईन आहेच मॅम.
ओके ओके.
येस.
वेलकम मॅडम.
Thank you, ma'am. Good afternoon, all.
आपण चालू करतो एक 5 मिनिटात मॅडम. जॉईन
ठीक
राहा.
आहे, no issues.
ओके ओके मॅडम.
Okay.
चौधरी सर, मकरंद चौधरी सर.
येस मॅम, चालू करू का?
हां, इंट्रोडक्शन द्या सर.
Yes. Yes. Good afternoon, all of you, and welcome all of you. Tapasvi Public Charitable Trust, Yevti's Venkatesh Madan Senior College, Dharashu, Department of Microbiology, in collaboration with Microbiologist Society of India, under PM Usha scheme, that is Pradhan Mantri Uchchatar Shiksha Abhiyan, organizes 2 days state level workshop. In this workshop, in the first day, this is the last session. May I welcome to Dipti Dhanraj Jadhav ma'am, API's Atlas LLP Gujarat. The designation of ma'am's team leader and working place is Ahmedabad. Now this is a short introduction about ma'am's. Ma'am have completed her bachelor degree in B Pharma from Indira College of Pharmacy, Pune. After that, she joined API Atlas LLP in 2022 as data analyst. Based on her ability and skill, they promoted ma'am as a template HR coordinator, and with achievement and contribution, summarize experience and accomplishments to highlights for a higher level position as a team leader. Also, she have been delivering guest lectures on different topics of pharmaceutical departments. May I request ma'am to start her guest lecture, ma'am. Please, ma'am.
Thank you so much, sir. May I start now?
Yes, ma'am.
Yeah.
You will start, ma'am.
The screen is visible.
Yes, ma'am. This is visible.
Okay.
Yes.
Today we are going to start the introduce about the case number and formulation. The key points are given below. What is case number and HSN code? Second one is what is raw products and products? What is the pharmaceutical APIs and intermediates, impurity in products and therapeutic categories? So first of all, we know about the case number. Anyone know about the case number? Hello, am I
Yes,
audible?
ma'am. Yes, ma'am.
Okay. Cash number is nothing but chemical abstract services. It is an unique identification number which is provided to identify any chemicals in the pharmaceutical industries. It designates only one substance, has no chemical significance. A cash registry number is a numerical identifier that can contain up to 10 digits, divided into happens into 3 parts. For example, paracetamol. Paracetamol has the cash number is 103 90 2. Second one is HSN code. HSN code is harmonized system code, which is basically used for the differentiate the 1 to 98 chapters of chemicals, like 1 to 98 chapters. Chapter 28 is for the inorganic compounds, chapter 29 is for the APIs and organic compounds, and chapter 30 is for the formulations. It is 7 digits code. Raw products. Raw product is the chemical name of the pharmaceutical substance or the pharmaceutical products. It can be identified as a chemical substance or API. And API is basically organic or inorganic. Organic and inorganic compounds are the chemical substance which is identified by their structure, appearance, and state. The examples of chemical substance that is organic 2-methyl 5-acetic acid. Second one is paracetamol. Pharmaceutical APIs. Active pharmaceutical ingredients that is nothing but APIs. APIs are the active components in the pharmaceutical drugs that produce the required effect on the body to treat the conditions. The active ingredients which contains in medicine, example, an active ingredient which is used in relief pain included in painkiller, that is aclofenac, diclofenac, ketorolac. These are the examples of API. Sorry. Next one is intermediate. The intermediate is the chemical compound which is the process of becoming an API from raw material. It is called as intermediates. Pharmaceutical intermediates used in the manufacturing various types of the pharmaceutical ingredients. Example, emanative intermediates. Second is 2-methyl-5-nitroaniline. So which language is comfortable for the students? Only English or Hindi may be used?
Yes, ma'am. Both आप use कर सकते हैं, ma'am. You have to use both language, English and Hindi also. Yeah.
ठीक है. इंटरमीडिएट्स जो रहते हैं, जो कि कौन सा भी एपीआई हम लोग जब मैन्युफैक्चर करते हैं, उस बीच में जो केमिकल आता है, जो कि वो प्रोसेस कंप्लीट करने के लिए यूज किया जाता है, दैट इज नथिंग बट इंटरमीडिएट. लाइक एग्जांपल, हम लोग जब भी कुकिंग करती है उस टाइम पे वहां पे जो साल्ट यूज किया जाता है, उसके बिना कोई भी खाना कंप्लीट नहीं होता है. तो वैसे इंटरमीडिएट भी वहां पे वैसे काम करता है. Second one is impurity in product. Impurity is defined as the foreign particles that affect the purity of pharmaceutical substance. Foreign particles that shows the adverse effect. Example, heavy metals, leads. Second, impurities that reduce the effect of active substances. Any questions? Next one is the therapeutic categories. Drugs are the classified into different groups according to their chemical categories and structures, how they are used to treat the significance disease. Next one is the hello. Example, analgesic, antibiotic, and anticancers. There are some websites or the references to find out the cache number, PubChem, Chemical Books, Sigma Aldrich, Clearson, Simpson Pharma, and Pharma Affiliates. Anyone questions about the cache number? Related to cache number, anyone has questions?
Yes, student. Can have any questions?
Atorvastatin 40 mg. That means the products contain atorvastatin, that is API, and having strength 40 mg, which shows the pharmaceutical effect. Second is API. Active pharmaceutical ingredients has the components, drugs to produce required effect on the body to cure type of condition. The active ingredients which contains in medicine which shows the pharmaceutical action under the body. Example is painkiller, that is NSAIDs, we can call as ketorolac and aspirin. प्रोडक्ट टाइप इन द फॉर्मुलेशन. प्रोडक्ट टाइप को हम लोग डोसेज फॉर्म भी कह सकते हैं, जो कि हम लोग एपीआई के द्वारा टैबलेट्स, सिरप्स, इंजेक्शन, ये सब इसका हम लोग मैन्युफैक्चर करते हैं, उसको प्रोडक्ट टाइप बोल सकते हैं हम लोग. द प्रोडक्ट टाइप रेफर्स टू द फिजिकल फॉर्म और द डोसेज फॉर्म ऑफ द फाइनल प्रोडक्ट. इट डिस्क्राइब्स द वे द फार्मास्यूटिकल इंग्रेडिएंट्स is presented to be patient. There are main three types of product types, that is in the form of three states, that is solid, second is semi-solid, and liquid. The example of the solid, that is tablet, then caplets भी आते हैं उसमें. Second is semi-solid, creams, ointments, and third one is liquid. The example of liquid product type is syrup and suspension. The examples of the product types are which are given added to our projects and to our formulation, that is tablets, injections, capsules, cream, drops, suspension, gel, inhaler, ointments, infusion, powder, vaccine, jelly, oil, spray, lozenges, lotion, balm, granules, suppositories, capsule, caplets, pellets, emulsion, pills, patches, elixir, and aerosols. These are the examples of the product type. Then what is the ETC category in the formulation? The ETC category is the anatomical therapeutic category or anatomical therapeutic chemical is the category is the classification system used to the group medical products according to their therapeutic effect, mode of action and anatomical targets. Foreign particles, unwanted, extraneous and extraneous that are present in the medical products, medical device, biological sample that show the adverse effect. Here are some anatomical therapeutic chemicals categories are given below, that are A to V are main categories, that is A, that is alimentary tract and metabolism, B is the for blood and blood forming organs, C is for cardiovascular system and D for dermatologists, G genitourinary system and sex hormones, systemic hormones preparation including sex hormones and insulins. Next one is J, anti-infectious systemic use. L is for anti-neoplastic and immunomodulating agents. M is for muscular skeletal system. N, neurosystem, and P, parasitic products, insecticides and repellents. R for the respiratory system, and S for the sensory organs, and final one is for the various. Here are some examples, that is BD-Fine. It is cardiovascular systems. Example is main category. Second is for betamethasone. Its ATC category belongs from the dermatologicals. Third one is for combinations also there are given ATC categories, like combination is given below as the cefiximide, doxycyclines and lactobacillus. These three APIs combinations are used to treat like general anti-infectious systems, that is their ATC category. मॅम कोणाला काही क्वेश्चन्स असतील तर त्या रिलेटेड आपण लेक्चर एक्सटेंड करू शकतो, म्हणजे मला माहिती देता येईल त्यांना. मी आता क्लिअर करू शकते, पण त्यांच्या डोक्यामध्ये काय क्वेश्चन्स येतात ते मला क्लिअर करता येईल. हॅलो, एम आय ऑडिबल?
Yes.
ऍक्च्युली सर शीट जी आहे प्रेझेंट केलेली आहे, मी जे काही सांगितलेलं आहे, पण त्यानुसार लॉजिकली काही क्वेश्चन समोरच्याला येऊ शकतात, त्यानुसार आपण त्यांना समजून सांगू. डिरेक्टली सांगितलं तर त्यांच्या डोक्यात काही क्वेश्चन्स येण्याची वेळ येऊ शकणार नाही त्यांना नंतर.
Okay. Yes, ma'am. Yes, from student side, anybody is there who is having the question? Yes, ma'am. My question is there. The API means to active pharmaceutical ingredients. Madam, any bad effect is having from APIs? Likewise, if we see the yes, ma'am.
Yes, if you take it in excess amount, it may shows the adverse effect on it. मतलब हम लोग जब एक्सेस अमाउंट में कोई भी चीज ले लेते हैं तो उसका एडवर्स इफेक्ट हो ही जाता है. एडवर्स इफेक्ट इसका भी होता है सर कि एलर्जिक भी होता है, कई लोगों को सूट नहीं होता है. बाहर
Yes, yes. Student from student.
What is the process doing MSc Biotechnology in other countries?
Hello.
Madam, hello.
Ma'am,
Huh?
question is
Yes. Student, one by one, you have to ask the question.
हेलो मैडम.
हाँ.
हेलो मैडम, एपीआई क्या होता है मैडम? एक डाउट था आपसे.
एपीआई इज अ मतलब ऑर्गेनिक भी प्रोडक्ट है. सर इको हो रहा है वॉइस, किसका डबल माइक चल रहा है?
Yes, student, you have to.
एपीआय हा जो
Yes.
सर मराठी सांगू शकेल मी यांना?
Yes,
म्हणजे
ma'am. चालू शकतो.
दोन्ही
मराठी
सांगते.
चालू
हो.
शकतो. हो, मराठी चालू शकतो.
एपीआय हा जो पदार्थ आहे तो ऍक्टिव्ह फार्मास्युटिकल इन्ग्रेडियंट्स आहे. म्हणजे आपण जी काही टॅबलेट सिरप घेतो त्याच्यामध्ये तो कमी प्रमाणात असतो. त्याची स्ट्रेंथ बरोबर असते. जेव्हा आपण घेतो आपल्या बॉडीमध्ये फार्माकोडायनामिक आणि फार्माकोकायनेटिक्स इफेक्ट शो करतात. पहिल्यांदा आपण टॅबलेट घेतो, त्यानंतर ती ऍब्झॉर्ब व्हायला सुरू करते, त्यानंतर ती मेल्ट व्हायला सुरू करते. ऍब्झॉर्प्शन मेटाबॉलिझम प्रोसेस झाल्यानंतर ती ब्लड मध्ये सर्क्युलेट होते आणि मग त्याचा फार्मास्युटिकल इफेक्ट शो होतो. त्यासाठी आपले डोसेज फॉर्म पण वेगळे वेगळे दिलेले आहेत. टॅबलेट थ्रू घेतलं आपल्याला एपीआय तर त्याचा इफेक्ट थोडा उशिरा शो होतो. आपण कधी पण बघतो ज्यावेळेस आपण आयव्ही और इंजेक्शन थ्रू जे काही आपण एपीआय घेतो त्याचा इफेक्ट लवकर होतो. त्याचं कारण जे आहे फार्माकोकायनेटिक आणि फार्माकोडायनामिक इफेक्ट आहेत. ऍब्झॉर्प्शन, मेटाबॉलिझम, डिस्ट्रीब्युशन अँड इलिमिनेशन या सगळ्या प्रोसेसचा डिफरन्स येतो त्याच्यामध्ये. जेव्हा आपण इंजेक्शन और आयव्ही थ्रू जेव्हा घेतो डायरेक्टली ब्लड मध्ये ते डिस्ट्रीब्यूट होऊन जातं, पण जेव्हा आपण टॅबलेट थ्रू घेतो त्या प्रोसेसमध्ये वेळ लागतो.
Yes.
क्लिअर आहे? नाही तर आपण परत दुसऱ्या भाषेत सांगू.
येस मॅडम, येस क्लिअर आहे.
ठीक आहे. एनी मोर क्वेश्चन्स?
विद्यार्थ्यांचे क्वेश्चन आहेत का काही? असेल तर प्लीज लवकर विचारा.
सर हम लोगों ने कैश नंबर का अभी बताया कि कौन से भी केमिकल का एक यूनिक आइडेंटिफिकेशन नंबर है वो, जो कि गवर्नमेंट ने वो अप्रूवल दिया हुआ है. हर एक केमिकल का अलग होता है, जो कि रेगुलेटरी अफेयर्स पता है सब लोगों को. इसमें कौन सा भी ड्रग हमें एक्सपोर्ट और इंपोर्ट करने के लिए ना उसकी रेगुलेटरी अथॉरिटी का रजिस्ट्रेशन करना जरूरी होता है. उस टाइम पे हम लोग ड्रग सेफ्टी मटेरियल शीट बनाते हैं. उसमें जो भी केमिकल्स यूज किया जाता है, एक्सिपिएंट्स हो, एपीआईज हो, बाइंडिंग एजेंट्स जो भी है, उसके सामने उसका कैश नंबर मैंडेटरी होता है. वो यूनिक आइडेंटिफिकेशन होता है. एक क्वेश्चन आपको आना चाहिए था, जो कि है पैरासिटामोल. उसका सिनोनिम आता है. सिनोनिम मतलब उसको एसिटामिनोफेन भी कहा जाता है. तो आपको एक क्वेश्चन नहीं आया कि एसिटामिनोफेन और पैरासिटामोल का कैश नंबर यूनिक आ सकता है या डिफरेंट आ सकता है, सेम आ सकता है? हेलो.
Yes, ma'am.
हाँ, ये क्वेश्चन किसी के दिमाग में आया नहीं. मतलब ये सबका अलग अलग कैश नंबर रहता है, तो जो सिनोनिम्स है उसका भी अलग रहेगा, सेम रहेगा. मैंने अभी एक बताया कि रेगुलेटरी अफेयर्स जो भी है उसका रजिस्ट्रेशन करना पड़ता है. हर एक कंट्री के गाइडलाइंस अलग होते हैं. उसके टाइप से जो भी इंग्रेडिएंट्स रहता है उसका हम लोग वहाँ पे कैश नंबर ऐड करके सब इन्फो ऐड कर देते हैं. ठीक है, अभी फॉर्मुलेशन आया। जो भी है उसका रजिस्ट्रेशन करना पड़ता है, हर एक कंट्री के गाइडलाइंस अलग होते हैं। उसके टाइप से जो भी
सर म्यूट करने के लिए बोलो, इको हो रहा है वॉइस।
टेस्ट नंबर ऐड करके सब इन्फो ऐड करना पड़ेगा। ठीक है, अभी फॉर्मुलेशन
चौधरी मॅडम तुमच्या याच्यावर इको आले. चौधरी मॅडम.
लाईक एक्झाम्पल डोलो 650 टॅबलेट मध्ये जो एपीआय यूज केलेला आहे तो आहे पॅरासिटामॉल. त्याच्यामध्ये स्ट्रेंथ ब्रँड नेम मध्येच आपल्याला समजलेलं आहे की 650 याच्यामध्ये स्ट्रेंथ दिलेली आहे, म्हणजे जे काही एपीआय आहे ते किती क्वांटिटी मध्ये घेतलेलं आहे आणि बॉडीला किती गरजेचं आहे त्यामध्ये दिलेलं आहे. मग त्याच्यामध्ये पॅरासिटामॉल एचसीएल त्यामध्ये हे केलेलं आहे, आपला एक्सिपियंट्स गेलेला आहे, ऍडिटिव्ह साल्ट म्हणून गेलेला आहे. तर आपल्याला त्याचा पण कोड एकच येईल का तो पण डिफरंट येईल, कोणी सांगू शकेल? सर स्टुडंट्स ऍड आहेत की फक्त फॅकल्टी आहे सर?
स्टुडंट पण आहेत मॅडम. नियर
हा,
अबाउट
स्टुडंटला
पार्टिसिपेट.
ओके. स्टुडंटसाठी पण हाच क्वेश्चन आहे. पॅरासिटामॉल एचसीएल आहे, त्याचा युनिक कॅश नंबर जो आहे तो सेम येईल का फक्त पॅरासिटामॉल आणि एचसीएल चा वेगळा वेगळा येईल?
हॅलो मॅम.
येस
मॅम मे बी म्हणजे जे आता तुम्ही डोलोचं एक्झाम्पल दिलं त्यामधलं पॅरासिटीमॉलचं तेच केस नंबर राहील आणि म्हणजे जेव्हा ते त्याच्यामध्ये ऍड होईल तेव्हा त्याचा डिफरंट केस नंबर होईल.
ओके, राइट. आप लोगों का भी इतना ही ध्यान रखना है, कैश नंबर का जब प्रोसेस हम लोग करते हैं, आपको जब पैरासिटामॉल का कैश नंबर पूछा जाए, सिर्फ पैरासिटामॉल का वहाँ पे हम लोगों को आइडेंटिफाई करना है, मतलब गूगल से मिल जाएगा, आप लोगों को साइट दिखाई गई है वहाँ पे. जब पैरासिटामॉल एचसीएल का पूछा जाएगा तभी वो अलग रहेगा, ठीक है? जस्ट मिनिट सर.
हॅलो
यस.
मॅम, कॅश नंबर फक्त केमिकल प्रोडक्ट्सलाच व्हॅलिड राहील का का बायोलॉजिकल प्रोडक्ट्स आणलो आपण मग त्याला पण कॅश नंबर द्यावा लागेल, लाईक इन्शुलिन वगैरे?
इन्सुलिनचा पण आहे कॅश नंबर.
ते बायोलॉजिकल मध्ये येईल असं होईल?
हर एक केमिकल, हर एक प्रोडक्ट, बायोलॉजिकल प्रोडक्ट असो कोणताही असो त्याचा कॅश नंबर दिलेला आहे. एनीवन नोज द इंसुलिन हाउ प्रिपेयर्ड, मतलब कैसे प्रोड्यूस हुआ है?
फ्रॉम E. coli.
Yes, right. अभी मैंने बताया कि जो भी ड्रग रहता है उसका फार्माकोडायनामिक एंड फार्माकोकाइनेटिक इफेक्ट्स होते हैं. फार्माकोकाइनेटिक्स क्या होता है? जो स्टडी है, मतलब जो इफेक्ट ऑफ द ड्रग ऑन बॉडी, मतलब हमारे बॉडी पर जो इफेक्ट होता है ड्रग का, और उसका मैकेनिज्म, ड्रग एक्शन जो रहता है उसके बीच का रिलेशनशिप बिटवीन द ड्रग एंड कंसंट्रेशन इफेक्ट, उसको हम लोग फार्माकोडायनामिक्स बोल सकते हैं. Pharmacodynamics is often summarized as the study of what drug does to the body. Clear? म्हणजे फार्माकोडायनामिक काय आहे? आपला ड्रग जो काय आपल्या बॉडी सोबत काय इफेक्ट करतोय. आणि फार्माकोकायनेटिक म्हणजे जी बॉडी आहे ड्रग सोबत काय रिॲक्शन देते त्याला आपण फार्माकोकायनेटिक म्हणू शकतो.
Yes, ma'am. Please continue, ma'am. I
Yes,
think there
yes.
is no question, ma'am. Yes, ma'am. Yes.
Yes. Pharmacokinetics is the quantitative study of the drug movement in through the out of the body. म्हणजे जेव्हा आपण टॅबलेट घेतो आणि त्याचं इलिमिनेशन होईपर्यंतची जी प्रोसेस आहे त्याला फार्माकोकायनेटिक्स म्हणतात. इट इज द स्टडी ऑफ द प्रोसेस बाय विच द ड्रग absorbed, distributed, metabolized and eliminated by the body. The absorption, distribution, metabolism and the excretion of the drug all involves its passage across the cell membrane. Pharmacokinetic properties are the affected by the route of administration and the dose of administration drug. What are the process of the drug formulation? Anyone knows?
Are you projecting your screen, madam?
No, ma'am.
Is there any PPT? Is there any screen sharing?
मैम इससे पहले पीपीटी शेयर किया गया था, उसके बेसिस पे मैं अभी इनको क्रॉस क्वेश्चंस करके फिर बता रही हूँ बाकी का.
Projecting your screen, madam.
No, ma'am.
Is there any PPT? Is there any screen
हेलो. इससे पहले पीपीटी शेयर किया गया था, उसके बेसिस पे मैं अभी इनको क्रॉस क्वेश्चंस करके फिर बता रही हूँ बाकी का.
Okay, okay, madam. Continue.
Okay. ड्रग फॉर्मुलेशन प्रोसेस करते समय जो स्टेप्स दिए जाते हैं, फर्स्ट ऑफ ऑल डिस्कवरी एंड डेवलपमेंट उसका किया जाता है, देन उसकी प्री क्लिनिकल स्टडीज की जाती है. प्री क्लिनिकल स्टडीज बेसिकली वो एनिमल्स के ऊपर किया जाता है. इफ एनिमल रिस्पॉन्स पॉजिटिवली देन इट मार्केटेड एंड discovered in the market. Then third is the clinical research, fourth is the FDA drug review, and five is the FDA post-market drug safety monitoring. Okay. The more about the discovery and development of the drug formulation process, that is typically research discovering new drug through new sites and the new sites into the disease process that allow the research to design a product, stop or reverse the effect of the disease. Many tests of the molecular compounds to find the possible beneficial effect against any of large number of diseases. Existing treatments have a unappreciated effects. New technologies such as those that provide new ways to target medical products, specific sites within the body or manipulate genetic material. At this stage in the process, thousands of compounds may be potential candidates of the development as a medical treatments. After the early testing, however, only a small number of the compounds look uh, promising and they call for the further study. Development. Once the researchers identify the promising compound of the development, they conduct experiments to gather information on the how it is absorbed, distributed, metabolized, and excreted. It is potential benefit and the mechanism of the action. Best doses, the best way to give the drug such as a mouth or injection. मतलब ओरल और आईवी दो रूट से हम लोग दिए जा सकते हैं. ठीक है. साइड इफेक्ट एंड एडवर्स इफेक्ट दैट कैन ऑफ्टन बी रेफर्ड टू एज टॉक्सिसिटी. How it is affects different groups of the people such as by gender, race or ethnicity or differentiation. How it is interacts with the other drugs and treatments, its effectiveness as the compare with similar drugs. मतलब ये जो डेवलपमेंट एंड डिस्कवर जो रहता है उसकी मेटाबॉलिज्म डिस्ट्रीब्यूशन एक्सक्रीशन एंड पोटेंशियल बेनिफिट्स ऑफ द मैकेनिज्म एक्शन, उसके द्वारा हमें ये डेवलपमेंट करते समय उसकी स्टडी करनी पड़ती है। उसका डोसेज फॉर्म कैसे दिया जा सकता है, स्ट्रेंथ कैसे दी जा सकती है, इसका सबका स्टडी करके ये फर्स्ट प्रोसेस की जाती है। Okay. Second step is for the preclinical research. Before testing a drug in people, researchers must find out whether it has the potential to cause serious harm, also called as toxicity. The two types of preclinical research are in vitro. FDA requires the research to use good laboratory practices, define a medical products development regulations. For the preclinical laboratory studies, the GLP regulations are found in the 21 CFR parts. These regulatory sets the minimum basic requirements for the study conduct, personal facilities, equipment, written protocols, operating procedures, study reports, and the system of the quality assurance oversights for the each study of to help assure a safety of FDA regulates products. Usually, preclinical studies are not very large. However, these studies are must provide detailed information on the dosaging and toxicity of the levels. Are after the preclinical testing, research review their finding and decide whatever the drugs should be tested in the people. मतलब प्रीक्लिनिकल स्टडी जो होता है, जो एपीआई है और जो ड्रग फॉर्मुलेट किया हुआ है, उसकी टॉक्सिसिटी और हार्मनेस चेक करने के लिए प्रीक्लिनिकल स्टडी की जाती है। Third one is clinical research. While preclinical research answers basic questions about the drug safety, it is not a subtitles of the studies of the way the drug will interact with the human body. Clinical research refers to study or trials that are done in people as developers design the clinical research phases and begins the investigational new drug process, that is IND. Process they may go through the before clinical research began. That is designing clinical trials, research design clinical trials to answer specific research questions related to a medical products. These are the trials follow specific study plan called protocol that is developed by researcher or manufacturer. Before clinical trials begins, researcher review prior information about the drugs development.
Ma'am.
Yes.
Yes, sorry for the interruption, ma'am. This is
Yeah.
live YouTube, ma'am. So please, you can start your video.
Yeah, sorry.
Video camera, ma'am. Yes, ma'am. Because this is live YouTube, ma'am.
Yeah, sorry.
Yes, ma'am.
Now visible, sir?
Yes, ma'am. Yes.
Before the clinical trials begins, the researcher review prior information about the drugs and development research questions and objectives. Then they can include who qualifies the participate section of criteria. Second is how many peoples will be part of the study, how long the study will last. Then whatever there is a group control group and the other ways limit research bias, and how the drug will be given to the patients at the what dosage, what is assessment will be conducted, when and what data will be collected, how data will be reviewed and analyzed. Clinical trials follow a typical series from the early small scale phase 1 studies to late stage large scales and phase 3 studies. The investigational new drug process. New drug process must they submit investigational new drug application FDA before beginning clinical research. मतलब जो भी clinical research करना चाहते हैं उससे पहले उनको IND application करना पड़ता है जो कि FDA के थ्रू लिया जाता है. That is Food Drug Administration. In the NDA application, developers must include animal study data and toxicity, that is side effects that cause the great harm data, manufacturing information, clinical protocols for the study to be conducted, data from any prior human research and information about investigators. Okay. Fourth stage is the FDA drug review, that is Food Drug Administration review. If the drug developer has the evidence from its early test and preclinical and clinical research that drug is safe and effective for its intended use, the company can file the application to market the drug. मतलब ये जो FDA होता है इसको हम लोग जब भी रजिस्ट्रेशन करते हैं उसका एक स्ट्रॉन्ग एविडेंस होना चाहिए कि इसका कोई टॉक्सिसिटी नहीं है, कोई साइड इफेक्ट नहीं है, जो भी हम लोग ये मार्केट में सेल करना चाहते हैं जो भी प्रोडक्ट्स, उसके लिए ये सब प्रोसेस करनी पड़ती है. The FDA review the team thoroughly examines all submitted data on the drug and makes the decision to approve or not approve. उसके बाद हम लोग ने जो भी प्रोसेस किया है उसका एप्लीकेशन एंड डिटेल्स शेयर करने के बाद FDA अभी उसका अप्रूवल करेगी या ना करेगी, अभी FDA टीम उसको क्रॉस वेरीफाई करके हमें एप्लीकेशन का रिजल्ट दिया जाता है. उसमें रहते हैं कि न्यू ड्रग एप्लीकेशन. A new drug application tells the full story of drug, its purpose to demonstrate that drug is safe and effective for intended use in the population studies. A drug developer must include everything about the drug preclinical data to phase 3 trial data in NDA. Developers must include reports on all studies that is analyzed along the clinical result. Developers must have included proposed labeling, second, safety updates, drug about information, patient information, any data from studies that have been conducted outside from United States. Second is institutional review and board compliance information, then direction of use. Last stage is FDA, post-marketing drug safety monitoring. Even through clinical trials provide important information on a drug's efficacy, safety, it is impossible to have complete information about the safety and the drug time of approval. Despite the rigorous step in the process of the drug development, limitations exist. Therefore, the true picture of the products safe actually evolves over the months and even years that make a product's lifetime in the marketplace. FDA review reports of the problems and prescription and the over-the-counter drugs and can decide the cautions to dosage or uses of information as well as the measures of the serious issues. Any questions? Any questions, guys? Hello, Makran sir, am I audible?
Yes, ma'am. Yes, please.
Any questions from yourself or from students?
Yes, this session is open for all. This is online and offline who are joined there.
Yeah.
We can ask the question. Yes, student, from student side, any question is there? You may ask.
Does government give approval to do preclinical and clinical testing?
उसकी रजिस्ट्रेशन भी करनी पड़ती है। प्री क्लिनिकल ट्रायल के हमें एक सेटअप प्रॉपर सेटअप होना चाहिए, एक्सपर्ट्स भी लगते हैं जो प्री क्लिनिकल ट्रायल्स कर रहे हैं। वो जो एनिमल्स है वो भी गवर्नमेंट से हमें प्रोवाइड करने मतलब परचेस करने पड़ते हैं, उनके ऊपर कुछ हार्म नहीं होना चाहिए, सब कुछ रजिस्टर्ड रहता है, फिर हम क्लिनिकल ट्रायल्स उनके ऊपर कर सकते हैं।
Can you please explain about computer-aided drug designing?
What?
Computer-aided drug designing.
No, I'm not part of that designing part.
Okay, ma'am.
Hmm. Any more questions?
Yes, student. Anybody is having question? I think, ma'am, there is no question. I hope
Okay.
there is no more question, ma'am, from our side.
Okay.
Yes, this is the conclude session. On behalf of Venkatesh Mahajan Senior College, Dharashu, I extended my heart-left gratitude to Mrs. Dipti Zadhu, ma'am, for her insightful and informative session on case number EPS Pharmaceutical. Ma'am, we truly appreciate the time and effort you took share your knowledge with us today. Your expertise and experience in this field are invaluable, and we are grateful for opportunity to learn from you. I would also like to extend my thanks to organizers and, of course, a big thank to our audience students for your participation and engagement. Once again, thank you and very much.
Thank you so much for guys
Yes, ma'am.
supporting. Yeah.
Thank you. Yes.
Thank you.
Over to Chaudhary, ma'am. Chaudhary, ma'am. Yes. On behalf of Venkatesh Mahajan Senior College, I thankful all of you on this auspicious day to organize an online session. This is a fourth session, and I thankful to all organizing committees, student, participant, offline, online. And there will be a offline session
Hello.
whole day tomorrow. Yes, ma'am. Chaudhary, ma'am.
This is a fourth session, and I'm thankful to all organizing committees, students, participants, who are involved. There will be
Yes, sorry, Chaudhary, madam. Are you hearing?
Yes, sir. Yes, sir. Just a minute.
Yes,
Let me
ma'am. Yes.
conclude, sir.
Yeah.
Hello. Am I? Huh. Yes, sir. As a organizing secretary, I am coming here. Today's sessions, though these are the lengthy sessions, I thanks to all participants for their sincere presence during all the sessions. Also, I thankful to the guest speakers of today's online sessions. Tomorrow, the schedule is totally offline schedule and every participant must be present physically here in Venkatesh Mahajan College, Dharashiv, and filling feedback form is compulsory to all, which give your guideline about today's sessions, and it is a mode of presentity and attendance for you throughout the all 4 sessions. So, thanks to all, and I conclude today's session here. We will meet tomorrow in at 10 a.m.
Okay, ma'am.